So this is in the case of chlamydomonas. In the plant, PSAI is chloroplast encoded, but some peripheral subunits are not completely conserved. So PSA A and B chloroplast encoded and homologous polypeptide. So very similar, but it's not exactly the same. So these two subunits are migrated in a similar position, so you cannot distinguish two subunits are co-migrated. And this is very hydrophobic protein, 11 hydrophobic helices, transmembrane helices. So that's why this protein is, if you separate on the gel, the show the diffuse band. This is one of the characteristics of the very hydrophobic proteins. And also there are many relatively smaller subunits, sharp band, there's so many, and very small subunit. And 2 PSAI and J are very small, about 4 kilodalton. So it's usually difficult to separate on this gel. So we detected this by immunoblotting using antibody. In this preparation, PSAN and O are missing because these two subunits are very weakly bound to PS1. So doing a purification, these two subunits are lost. Okay, you need to be a very special use a special condition to isolate PS1 containing these two subunits. So because this region, many subunits has a similar size, so some polypeptide is very closely migrated or overlapping. So it's difficult to say how many, but actually assigned by immunoblotting or mass spectrometry, there are 9 LSCA subunits. Usually higher plants, vascular plants contain only 4 LSC1. But Chlamydomonas may be adapted to the condition of lower light to grow. So, thank you. It's nice. So, it's the same as other green algae contain more LSC1 light harvesting to harvest more light. Okay, and there are several small PS1 subunits. So these are the separation pattern. This is a little complicated. For example, LSCA 635 is migrated very closely, like a broadband. And LSCA 7 and 2 migrated at the same position, and LSCA 8 is a little bit lower, but it's overlapping. LSCA 1 and 9 are overlapping, it's the same position, something like that, and L, H, E, G, C, K. So this is a, although this is used very high resolution gel, it's a long gel and very special content of buffer. So this is one of the very best separation for us, but still many subunits are separated very closely or overlapping. Okay, so this is a PS1 LH1 supercomplex. Yeah, okay. So first, we want to. I want to show you that how this 9 LHC1 complex are accommodated in this PS1 LH1 supercomplex in Chlamydomonas, because this part is very much similar from the cyanobacterial PS1 core and to vascular plant, algae also. So this is almost we know. Only some peripheral subunit some missing or additional subunit. So this part we focus on this region. So first, we want to know this 9 LSC1 is present in PS1-LH1 supercomplex stoichiometrically. Okay, you mean each subunit one copy of each LHC1 present in this supercomplex, or not. Some are more than one copy, or some are half copies and heterogeneously associated or not. So to determine biochemically, we labeled proteins of chlamydomonas with carbon-14 radioactive isotope. Carbon-14 is not often used recently, but you can uniformly label all the protein by labeling a carbon-14. Then you can estimate the amount of each polypeptide quantitatively, because staining, this is staining. The staining is not proportional to the size of proteins, because some amino acid stain better than the other. So depending on the amino acid composition, the staining is not equal. So from staining, you cannot estimate the stoichiometry precisely. So we grow the cells in the presence of carbon-14 acetate. Acetate is a carbon source, so we use the carbon-14 acetate in the medium. And then grow the cells 2-3 days. Then whole proteins are uniformly labeled with carbon-14. Then we isolate this one and separate polypeptide, then detect radio activity by X-ray film, or some more quantitatively by some intensifying screen, and then the machine estimate the intensity of the signal. Okay. So this is a result after. This is not staining. This is a autoradiography. So intensity, the signal indicate the intensity of the radioisotope, radioactivity. So the intensity of this band represents the amount of protein. So we can estimate amount of proteins in this complex. But as I told you that there are many non-LH1 and also PS1 terminate migrated this region. So some peptides are very close or overlapping. So it's difficult to estimate, right? So we use 3 different separation method system. This A is you can separate LHF4, 6, 3, 5, 7, 2, and 2. 2 is a little bit diffuse, and 7 and 8 and 2. So LHCA 1 and 9, PSAD and something like that. So you cannot estimate each of the LHC1 by this separation. Then we used a different separation. We changed a little bit the buffer system of the gel. So relative migration of polypeptide changes. Okay. So here, LHCA4 is here, the 6, and here 3 plus 5. This is 7 plus 2. This is 8, 9, 1. But PSAD and F are in the same, something like that. This, under this A condition, these 2 are separated, but here it migrated on at one single band. But this one is LHCA 1 and 9 was single band, but under this condition, separated at 2 bands. Something like that. And we could mostly separate it combining these 2, but still not perfect, because LHCA 7 and 2 still difficult. So then we did 2-dimensional electrophoresis. Okay, so first SS page and second SS page is under the different gel system. So LSCA4 is here, 3, 5, 6, and 7, 2, 8. 2 and 8, 2 and 8. So combining these three gel systems, we could almost perfectly separate and estimate each subunit. Okay, then this is a result. LSCA4, based on the PS1 subunit, LSCA4 is 1 copy. It's a different system, but it's almost 0.9 to 1.0, so it's almost 1 copy. LSCA6 also 1 copy. LSCA3, 1 copy. LSCA5, almost 1 copy. So there's some deviation. And second, this page 278 is separated, but the other system, it's 3 migrated together, so 3 copies, but these each 1 copy. LSCA1 is somehow overlapping with other subunits, so with 9. So LSCA9 is almost 1 copy, and LSCA1 was 2 copies. So it's not, everything is not 1 copy. And this is PS1 subunits, 1 copies, okay? And the different condition is 2 copies, but it's separated as a single. So PS1 subunit has 1 copy, and LSC1 subunit 1 copy, except for LSC1. This is 2 copies. So PS1 LSC1 supercomplex contain 9 different LSC1 polypeptide, but copy number is 10 because LSC1 is 2 copies. That's our conclusion.
So can you please explain this temperature differences? Like what is the role of this?
This one?
Yeah, yes, sir.
Okay. We use a high molarity tris SDS page. This is give us a good separation condition. But if you run the gel, low temperature, high temperature, separation changes we found. So they're using the same electrophoresis condition, but under the very warm condition, and in the cold room, then we could separate differently. For example, this band, LSA1 and 9 migrate as a single band, but in the low temperature, separated. But high temperature, PSA-D and F separated as 2 bands, but low temperature is separated as single band. So combination of this different condition, we could separate all these 9 and 10, 11 polypeptides that are migrating very close to each other. That's possible. So, okay. So this is a biochemistry. So
the low molecular weight, they are very close.
Low molecular weight is, you mean, there's a 2. This is we call the low molecular weight, but this is very low. This is a medium. So this region, there are many polypeptides. So we wanted to separate nicely this region. But others are separated very nicely. We couldn't estimate these two, too small. Okay, so we are focusing on LSG1. So then 10 LSG1 subunits and how they are organized in complex. Okay, so what we did was chemical cross-linking experiment. So you can cross-link between neighboring subunit by chemical cross-linker. Okay? So if the 10 kilodalton polypeptide is cross-linked to 20 kilodalton polypeptide, that product is 30 kilodalton. Okay? That's a system. So only neighboring polypeptide are cross-linked. That's so that you can estimate where these 10 LHC1 are present in PS1, LHC1 supercomplex. Okay? So you don't need to know very detailed, but this is a result. Sorry. We make antibody first against each of LHC1. The LHC1 is very similar proteins. No, they are 9, but they are similar. So if you make the antibody, generate antibody against the entire polypeptide, so then this antibody may cross-react to other LHC1. So, but for chemical cross-linking, we need a very specific antibody. So we use the synthetic polypeptide as an antigen. Okay, that's the region, the polypeptide sequence, amino acid sequence region that is unique in each. So it took many years until we get 9 different antibody against each of LHC1, but finally we could. So, okay, this is a little bit complicated, so you don't need to see everything, but for example, it's if you look at which one? This one. We use the LHCA3 antibody and PSAK antibody, okay? And minus, so this is an immunoblotting of using anti-LHCA3, and this is immunoblotting of using anti-PSAK. Okay? And minus is no chemical cross-linker, okay? Without the chemical cross-linking. And plus is after chemical cross-linking, okay? Then, if you look at the minus, oh, this is, okay. LHCA3 is here. And PSAK is a little bit weak here. Okay? And after cross-linking, we could see a new band, upper band here. And here. Okay? And the position, when we detect it with LHCA3 and PSAK, the position is the same. This means after cross-linking, LHCA3 and PSAK was cross-linked and separated this position. So that means LHCA3 and PSAK are neighboring. So we did as many as we can, okay? So many different combinations. Okay, other example is, for example, because sometimes the antibody cross-react with non-specifically cross-react. So you have to eliminate artifact. Okay, I think this is clear one. LHCA2, LHCA9, without chemical cross-linking, this is the original. This is original, okay? And after cross-linking, LHCA2 have shifted this 3 here, and another one here. And also LHCA9 is after cross-linking, there's additional new band here and a little bit here. So this means LHCl2 and LHCl9 cross-linked. And if you see additional upper band, that may mean the third polypeptide may be cross-linked very inefficiently. Okay, so these results.
So this cross-linking we are doing separately for each protein, or
Yeah,
it is in the cell?
we did many plots. Okay.
Yeah, like we take out the protein, purify it, and then cross-link, or like how are in the cell?
Sorry, we use the thyroid membrane most cases. That is that prevent artificially cross-linking. Because if you isolate PS1 and then cross-linked, you may see a cross-linking between different complex. To avoid this, we use the thyroid membrane most cases. Okay, this is a cross-linking, very classical method, but in a typical biochemical method. So this is the result. Okay, summary. For example, LHCl1 cross-linked with LHCl9. So this is our list. PSAL cross-linked with PSAO, something like that. Then we made a model. Okay. This is the PS1 viewing from the stroma side. Okay, stroma side. And then PSA-A and PSA-B. Green part is PS1 core subunits. F, J, N, O. This. And white circle is LHC1. So as I show you, LHCl2 and LHCl9 cross-linked like this. So, but from the other cross-linking, we accommodated that. 2 and 9 is located in a PSAG ILH side here as a dimer. And also LHCl1873 and PSAN is here. If you isolate PS1, PSAN is easily lost, but if you use a thyroid membrane, N is cross-linked this region, so be sure that N is around here. And this for LHC1 arrangement is very similar to vascular plant PS1. And 5, 6, 4, 1 is located the second layer of tetramer of LHC1. But this is dotted line is we could not definitely assign one of the two LHC1 here. But from the arrangement, we are sure that second LHC1 is located here. So tetramer, dimer. It's arrangement of LHC1. This is our biochemical characterization. Okay, so I think data of the location of PSAN is very precisely we could assign here. That is still remain completely determined, but I think this is one of the most reliable results. Okay, so is that okay? So this is a little bit the classical, traditional way of assignment. Then we could did determination of structure by using a cryo-electron microscopy. So maybe you know the cryo-electron microscopy. It's this can At a low temperature, the frozen sample analyzed. So by analyzing, we could determine all the subunit and also cofactors, pigments in the complex with very high resolution. Okay, so the question is our result by traditional and classical chemical linking by chemistry. This is correct or not? Okay. That was a very thrilling moment when we started this. Our result was completely correct or not. I don't know. It's, I think, okay, just continue a little bit. So here we isolate PS1, LH1 supercomplex biochemically and then analyze the particle. So I'm not specialist of cryo-electron. This is a collaboration with a specialist. So if you take a photograph of the particle, looks like this. So many small particles you can see. Okay? But this doesn't say anything about the structure, of course. But somehow the software pick up all the particle. This is particle. Can you see like a particle? And then I don't know how many, 100,000 or so many particles. And also, of course, in the particle on the this is on the grid, the frozen. So particle maybe not always like this. Some are like this. No, many different orientation. It's frozen. That is important. And then software pick up and then sort it to the similar size, similar direction, no orientation, and then average. No, so different particle like this, then overlap automatically. They continue hundreds of thousands particles. So these are different particle, how to say, different orientation and averaged. So this is flat and the side view or many different things. Already, if you look at here, there's some core and then two layers like this. But this is different angle, two layers and core, something like that. Then this, from this data, some software assemble this two-dimensional the picture into three-dimensional. This process I don't know. But this is the results. No, what's the difference from here to here? Okay. This is the view from the stroma side. Okay. This is core and LH1, two layers. You see? Something you can see here. No? 4 particles. 2 layers. Then after analysis, refinement. So maybe it's difficult to see something like this. This part here is PS1 core. Okay. And this part, LH1. And this is also LH1. LH1, LH3, 7, 8, 1. LHCA 5, 6, 4, 1. LHCA 2, LHCA 9 is tetramer, 2 layers of tetramers, and 1 dimer. Dimer is between the PSAG and PSA-Li, this region. So our result was correct. Very fortunately. We ourselves could confirm the results later, okay? Okay, so cryo-electron microscopy reveals a more precise structure. And also, this rod structure is transmembrane alpha helix. Okay. So I showed before the LHC 2 had 3 transmembrane helices, right? So this LHC1 also 3 LHC, transmembrane helices and a small hydrophilic small alpha helix is also present. It's a very overall structure is very similar to PS LHC2. Okay. But LHCA2 here is a little bit different. This contains 4 transmembrane helices. So this is what's new finding. Result. And this is a polypeptide and also cofactor, chlorophyll A and B and carotenoid. So this is only cofactors, pigments, like this. Yeah. So, well, all the region pigments are distributed, but it's two-dimensional, so no, it's stromal side and the luminal side. We cannot distinguish between them. But it looks like rather uniformly pigments are present. And so the green is the chlorophylls in PS1 core, and this yellow chlorophylls in LHC1, light harvesting complex. So this is the side view. This is the thyroglobulin membrane. And there are many transmembrane helices. That's why PS1, LHC1 present in the thyroglobulin membrane stably. Okay. Some units are stroma side exposed to stroma side, and some are a little bit exposed to luminal side. Okay. And LHC1 is inner LHC1 belt is tetramer, and the outer tetramer here, and also dimer, LHCA2 and 9 is here. Okay, something like this. And many pigments are present. So, structure of PS1 LHC1 supercomplex. Definition is like that. It's a PS1 reaction center, PSA A and B. And PS1 core means 2 reaction center plus peripheral PS1 subunit. The vascular plant PS1 LHC1 contains 4 LHC1. So PS1 core plus LHC1 tetramer. Okay? And chlamydomonas has 4 tetramer, 2 layers of tetramer. So total LHCA1 8 and 1 dimer here. So total 10 LHC1. So our biochemical data also was correct. Okay. So I was very happy to know that. Yeah.
To the previous slide.
Sure. Sorry.
To the previous slide.
This one.
Yes.
Could you use the microphone?
Presence of number of LLCs.
I don't hear very well. So
Yeah. Is
Okay.
it audible now? Yeah. No, I wanted to know, sir, in case of the plants, when we see that light harvesting complexes are relatively lesser than that of the algal systems, like Chlamydomonas. So when we see, what do you think, like the membrane biology, like particularly when we compare the plant chloroplast membrane, lipid membranes, and then algal lipid membranes and their surface area, how it is encompassing these more number of LHCs.
More LH1.
Yeah, like when
How?
we see here in yeah.
Yeah, there's a contact site, the inner and the outer. So interaction to stabilize this two tetramer is, for example, say the hydrophobic interaction or some hydrophilic interaction like hydrogen bond or ionic interaction. This order together of this many weak interaction finally stabilize this. Very stable.
So will it lead to the thylakoid width to be more in algal thylakoids compared to the plant thylakoids? Like, because there is a lot of space is there, surface area is there in the plasma membrane lipid bilayer. No, I mean to ask, like, if the thylakoid surface area, the size itself becomes a bit larger in algae compared to that of plant because of incorporation of more of proteins,
Yeah, of
LSCs.
course, it's important thing is this interacting contact sites. So, and also this how to say, inner and outer is the circle is the size is different. So,
Yes, sir.
but these are completely fit. But depend on the organism. So small change required for stabilize this structure. But we don't know yet what's the main key point. But it's
Yes.
right.
Yes.
Yeah, it's okay. Okay. So I think the time. So I still continue the next lecture. The Raja will take over. Then the following lecture today, I continue. So I just this, and also the next my lecture deals with assembly of this complicated structure. Of course, we don't know everything, but in part of the assembly process we analyze. So your comment or questions are welcome.
Since I have one doubt, so regarding this LHCs, there are 10 LHCs, right? So in PS1. So we all know about the LHCs in PS2, how they are going to affect it in terms of any stress. They are going to either do state transition from PS2 to 1. So yeah. So in that case, any remodeling of this PS1 LHCs will happen, or they still be the same?
LHC2 associate this region in Chlamydomonas.
Yeah.
2 LHC2 trimer associate.
Okay.
But depend on the organism, sometimes the 3 LHC2 trimer associate, but it depends on organisms that grow under the different light conditions. So if the organisms grow under the very low light condition, in general, they have a more light harvesting complex. So because probably chlamydomonas is growing relatively, how to say, low light condition in the soil of the some not exposed, just shade of the other plant. So probably this is laboratory strain, but it's originally chlamydomonas must grow. Probably they grew under low light conditions. So that's why it contains more light harvesting complex. And also under some condition, LSA2 trimer is present here. And also, of course, and there are many LSU2 detached from PS1 and PS2. So these LSC2 trimers may transfer excitation energy to PS2 or sometimes PS1. But we don't know exactly the old LSC2, how they behave. We don't know yet completely. We know only excitation energy transfer from the LSC stably associated with PS1 or PS2. So this part is rather flexible depending on the organism. So that is a kind of adaptation to different light conditions. Any other question?
Sir, slide of this one mole components with plots.
Even the block?
Yeah, initial slides.
So?
Back.
Back?
Yeah,
This one? Yeah.
So, like, how did you identify all the super complexes in PS1 LS?
Okay.
One?
Uh,
These are from like.
These are obvious because it's high molecular weight. Of course, we detected this polypeptide by antibody.
Yeah, but everything like.
Everything we assigned by immunoblotting.
Okay.
Because
based on
we
molecular weight.
have antibody. And also, in the beginning, we also analyzed by mass spectrometry.
Okay.
Cut this band and analyze the mass after digestion with trypsin. And because we know genetic information, so it's easy to assign. But these too small are only detected by immunoblotting because too small polypeptide is sometimes difficult to assign by mass spectrometry. Somehow.
So these characterization are based on the mass spectrum only. Then.
No, this immunoblotting, and this I think it's immunoblotting mainly because using antibody staining and then using antibody, so this band is specifically reacted with antibody against PSA-K. And sometimes this region is difficult, so we use the mass spectrometry. So not all the mass spectrometry, because mass spectrometry is very sensitive. So you have a overlapping protein like this. So then mass spectrometry data show that there are two LSG1 polypeptides. So we cannot distinguish which is which. But in Western blotting, very delicate difference in the migration position, we can distinguish between two overlapping proteins. So sometimes immunoblotting is more powerful than mass spectrometry.
Sir, this structural composition is similar in control and stress.
Sorry, so I don't.
This structural composition is similar in control and stress, or whether there is any remodeling happening during stress.
No, I don't understand.
Control and stressful condition.
Sorry?
During control and stressful condition.
Press
And the same or there is any remodeling happening. Remodeling of this. Remodeling.
If you cell growth and the different environment condition.
Remodeling happening.
Okay. The question is, if you grow the cells under different condition, like low temperature or high temperature, low light or high light, I haven't carefully analyzed so far, but it is said that LHC1 are very stably associated with PS1 core. So the amount and also localization of LHC1 are not changed. Probably that's true. But peripheral trimeric LHC2 are more flexibly shuttled. So depending on the growth condition, it may lost and it's associated or detached. It may happen. So this part are very stable, I should say. Okay,
Sir, what about the PS2 core?
PS2.
PS2 have any remodeling or possible?
PS2 core part is stable, but LSC2 is depend on condition, but it's there are many reports of PS2, LSC2 super complex, and some reports show that one trimer of LSC2 are present, and some report under milder condition, yeah, there are 2 or 3, and so the difference is ascribed to the difference of the growth condition or purification condition. We don't know exactly, but it's a kind of a competition of the number of trimeric LHC2. So if you report 1, and then others report 2, and others also report there are 3. So, okay, 3 is more fancy than 2 or 1, that kind of things. But they are seeing, I think, PS2 with 1 trimeric LHC2 or 2 trimeric LHC2, 3 trimeric LHC2, but they show maximum number of trimeric LHC2 in the paper. There may be more LHC2 with single trimeric LHC2, but they don't care very much in the beginning. They want to show that 3 is the largest for the publication. This happens. So this difference may due to the growth condition or isolation condition. That the isolation condition means it's an artifact, right? Okay, so this is very still remain to be determined. If we find a better, milder detergent, then we may have more PS2 preparation with more LHC2. That may happen. So it depends on the development of new detergent or new isolation technique. So it's biochemists sometimes not physiologists. So and also structural biologists also like protein they just compete with resolution. We have a better resolution than the other, something like that. But they don't care about the function or physiology of the protein. So this happens. Okay. So I stop.
Okay, right, thank you. So we'll felicitate Kazuma-san. Yesterday she missed. We have, we'll have all of, please come here. We'll take group photo again.
Again? Really?
So So this you like maybe much better. Yeah, this is good. Yeah, please. Can we have photo? Selfie. Thank you for coming. All of you join for group photo. It can fit in it. Yusuf, you can. Thank you. You like this one better? Okay, yeah, you can be like that. Yeah, it's a break now.
मतलब लेने
Chai.
हाँ डुप्लीकेट्स इंट्रक्शन
Tomorrow, Punamaka will come. So I have some meeting. Went to Padmaja Hotel. We came back after exploring the nightlife of the town.
ఇదేంటి ఇది దానిపైన రైట్ వద్దలేదు. Yeah, I think good morning. You know me, right? Is it okay? I can stand like this. So the topic is role of photosynthetic pigments in chloroplast biogenesis. So we'll be discussing about the pigments, what are the pigments and their importance also we'll discuss, and how they are placed in the photosynthetic membrane, that we'll discuss maybe another 40-45 minutes. So we all know that, I mean, the earth, when 4 billions years ago, there was no oxygen, I mean, there was no gases. You see the left panel where you have the earth was like this. So then started with, the air, you have the burning kind of thing. Then there was the gases has formed. Then what you see here at the moment right now, the green phase, whatever this one, all these 1/3 of the water, you have, and also the green belt, and the photosynthesis is evolved such a way that, the plant, we are all surviving because of the plants. So now you all know that why I kept this slide. Maybe you can also get anywhere in the internet. So we all like sun, right? So if you don't have sun, and you will be frustrated, and you may not be feel fresh. So that's what, when you do, most of the people, you do Surya Namaskar, right? Those are, this is somewhere in the river somewhere. This is like, to get something, a juvenile, I don't know, or thing, you can say that you get something freshness and you start the day with that. And the birds, they fly, if there's, sun comes, the birds fly, all animals will be very happy. And anyway, this, from sun, you are getting energy. You say that positive energy. It's mythologically, or it's true that, you get a positive energy. I will tell you a story, like most of the European countries, they get almost 6 months, they don't have sun. You know that, right? Especially Norway, Sweden, Russia, some part of Russia, many other, in France. What they do, if they don't have sun for 3-4 months, you will get not frustration, it's a kind of, I am not getting the word. That is the thing. But you may not be like feeling like, something like
Laziness.
energy, whatever. So laziness, whatever. What they do is they go for phototherapy. How you go for a gym and all, they do phototherapy, means light. The rooms will be full of light, which is mimicking to the sun, and they stay there just after the hours, after the working hours, they go and stay in the sun, like artificial sun. If they don't get whatever vitamins, whatever you think, whatever you think, so it will be a deficient, so they may have other complication with the one. So likewise, I said, all this sun is very important. Now what we are also now producing a lot of solar energy, and many more things we'll discuss. So now the basic photosynthesis, it started with anaxigenic. After this, the archaea, say that archaea bacteria, they started with anaxigenic photosynthesis, where they don't fix CO2, but they use organic stuff like H2S and gases, and produce the ATP through purple bacteria, non-sulfur bacteria, sulfur bacteria. These are the bacteria. Then after that, about 2.5 billion years ago, the first oxygen species, cyanobacteria, evolved. Then you could see all these one, the autotrophs. That means autotroph is nothing that can synthesize their own food. And so then, the basic reaction of photosynthesis, I mean, you all know that, I don't need to explain. So it requires solar, that means light energy, and whatever is the light energy will chemical energy that will need, you have a two cycles, I mean light dependent, light independent. Light independent is also called dark cycle or Kelvin-Bunsen cycle. The whatever ATP is produced, ATP and NADPH is produced from linear electron transport, that will be utilized in Kelvin-Bunsen cycle to produce the all biomolecules, essential biomolecules like carbohydrates, then it goes to all lipids, proteins and all be synthesized. That's what you could see here, the carbohydrates and of course the water molecule and oxygen. So now why plants absorb only in the visible region? So this is the electromagnetic spectrum. You have a gamma rays, X-rays, UV rays, and the region is called visible region, 400 to 700 nanometers. Then infrared waves and radio waves. These are the the electromagnetic spectrum where plants absorb only the visible region. So can any of you tell why plants absorb only visible region? Of course, they absorb in UV region also, but the UV region maybe damages the plants. So plants can the photosystem, not only plant, photosystems will be get, and they absorb. They have a photosynthesis, the receptors where they can take it, they can absorb. But however, they may be deleterious to the plant. Of course, other radiations also. I mean, we don't get other radiations, and so in electromagnetic spectrum mostly we get near UV and little bit infrared. So where plants can absorb, but they have a mechanism to they cope up or a protective mechanism. So why plants are green? So it takes Webster color. What we say that different wavelengths of visible light are seen by human eye as a different colors, but they absorb. But they can be also plants are green, but they are not green, right? The eye which we are seeing because of reflection. You could see because whatever the light it takes. Now it takes red light. How we are seeing it blue? I mean green color. So that is all because of the reflection. So this I don't want to discuss because it has been discussing many people. Yesterday also Das sir was discussing, but these are the chloroplast. So now we'll talk about these pigments. The topic it says pigments. So plants have evolved with the various kind of pigments. The pigments are these are called the major pigment is chlorophyll E. The all others are accessory pigments. That means they help transferring the energy from these to chlorophyll. So now I showed you this picture. So looks so beautiful, right? So now the green plants, other the plants, the pigments or the chlorophyll pigments will convert into the carotenoids. The carotenoids have many other pigments. I will discuss bit later. I mean we can tell later. So carotenoids have xanthophylls, zeaxanthin, anthocyanin, luteins, like several pigments. They have a different colors in plant depending on the plants. Based on the plant, they change the colors in different colors. So these now, I mean, if you those who are going, if you are somebody visit European countries or Canada or in US, it's a big tourist place now. Right now, I think it is the September, October. So people go and just to see this beauty of this nature. I mean, night so first it's green, then it convert into the yellow color. From that yellow, it convert into reddish, pinkish colors, various kind of colors. It changes because carotenoids convert into, once it is chlorophyll, convert into carotenoid. Carotenoids will be then carotenoid, many carotenoids, like xanthophyll, zeaxanthin, anthocyanin, lutein. It's a phase wise. The conversion will be 1 to 1. It takes days, few days. Then you see that the colors. So how we can measure? We measure the chlorophylls? Yes, just take organic solvents, acetone or anything, methanol, DMSO. They can dissolve. You just crush it, the leaves, or even the cells, cyanobacteria. They just grind it or mix it, mixture, blend anywhere. So you can get all these pigments. So then you measure a simple absorption spectra where they have wavelengths, different wavelengths, and you can characterize whether the chlorophyll. I mean, it's already standard. Standards are there, or you can also, the fixed wavelength is already been in the literature. From that you can identify all these peaks. Let's say this one. This is the chlorophyll A peak. Let's say this is the chlorophyll B, and this is the carotenoid peak. So likewise, you can understand the nature of nature means how quantity you can quantify it. So then the structure wise, these are the chlorophyll structure. A simple change in the groups of this, you can see here. It's simple. I mean, these are the pital ring, the heterocyclic compounds, and majorly magnesium is the one, determines the green, the nature. With the same one in human, what is that? It's same one, same structure. It will be iron will be there. So hemoglobin. So we will have a iron. So then it will be the same, almost same structure. So the simple change in this, the methyl group to aldehyde group, it changes the group. The wavelength, absorption capacity also, like 663 is a chlorophyll A, then it goes to chlorophyll B is 652. So there will be about almost 12 nanometers, the shift will be there. So now they almost, beta carotene, of course this aromatic ring, and zeaxanthin also, you see that zeaxanthin same ring, you have a additional OH group, and this will tell about the zeaxanthin structures. Of course, this chlorophyll C, there are plants, the most of the land plants have chlorophyll A, B, chlorophyll A, B. But there are bryophytas or lower organisms. They will have, those who stay in those who live in extreme conditions, like salt or high temperature, high altitude. They will have a different, the same carotenoid, chlorophyll pigments, and even different carotenoid pigments also, up to F, chlorophyll A, B, C, D, F, now pigments, and even now they identified G also, G. So, but it is only the little structural change, but they all looks green. And can we measure all of them? This is, you can see here, all the chlorophylls can be measured. Again, as use acetone, methanol, just simple. You can see the structural changes, a small change in the structure, chlorophyll A, B, C, D, E. I said, I can go with many these are the bacterial chlorophylls, and these are the land plants. What you have, chlorophyll A, chlorophyll B. These are also same structures. Only the pital ring, there is a functional group. One functional group change can make lot of differences. Like bacterial chlorophyll, they absorb in longer wavelength. That is about around 800 nanometer, 900, even 1100. Where our Dr. Yusuf will be talking about the topic on Saturday, right? Day after tomorrow. So he will describe a little bit more on how they can, So you can isolate all these one. Now here, the additional, there are two, the pigments, bacterial chlorophylls, and you see that it absorbs somewhere 800 nanometer. There is phycocyanin, allophycocyanin, phycoerythrin, where the cyanobacteria, they have these pigments. So we'll discuss about all those one later. Okay, so about these are the pigments. And I've been, Chiro Takashi, and other people, Abhidas were talking about more about structure and the composition, biogenesis. So what are the application? But now many of you know that application, but through photosynthesis, what kind of application we can generate. So you people can, looking at this one, maybe you may have your own ideas and your perception will change now. So this we know that anything, whatever we are eating, this is only because of plants. Plants are whatever, because it's a cycle, life cycle. The animal system also has to eat the plants, but that is again through photosynthesis only all this will you get it. So I kept this one to pollution controls. All the plants can control pollution. Wait, what is this one? This picture? Oh, fog in Delhi. But what is that one inside? India Gate. So maybe in Australia you have seen that like that, right? That color. So this is more of fog with pollution, smoky type weather. But if you have more plants, they will try to can reduce this one. So now the pigments, chlorophyll, carotenoid, anthocyanin, they looks like this. So now what are the importance with the pigments? This also, maybe they all know that I teach this one in the class also. So now I mentioned here phycobilisomes. It's not only phycobilisomes, any pigments, chlorophyll, anthocyanin, lutein, all of them have this kind of roles. So we can use this one in pharmaceutical industry, and they will be acting as anti-inflammatory, neuroprotective, hepatoprotective, hypocholesteric. All this will be helping this one. You know that Spirulina platensis, what is this? It's not algae, it is a cyanobacteria, blue-green algae. So, and chlorella, and there are many other, the cyanobacteria or algal system. Chlorella is algae, green algae. So people take it as a protein rich, and it also protect. It's proved already. The research has been, already, a lot of research has been, carried out in many places where all these functions they have. So for girls, most of your girls here, so all these one colors, lipstick, sun protecting eyeshadow palettes, all can be used by using these pigments, because the cyanobacteria phycobilisomes, they have three kind of pigments. These are the pigments, phycobili proteins. So it's like this color and bluish color, a light bluish color. These three colors, independently you can make whatever the colors you would like to have, and these are all highly anti-inflammatory and anti-diabetic also. So, and also you say that therapeutics, Alzheimer's, aging, I think I just missed with HRO if you have been there. So my students, I think most of my students visited Japan. So every meal they wrap with the cyanobacteria. Actually, the red algae are cyanobacteria. Sushi, Japanese food, sushi. What are other foods? Onigi, whatever you say that. With rice, they wrap with that. Any food, or they also use the seaweed, which has a highly abundance of the pigments, high pigments. It's all because of one. So now HRO and his wife, if you see them, how old they are, you can predict how old they are. 40 or younger than me. So anyway, so Japanese, Koreans, they live longer. Maybe one of the reasons every meal it comprises with the wrap with this, the cyanobacterial or red algae, which contains high amount of phycobilisomes. So you want to live longer, you can use these pigments. Okay, we discussed antioxidants, and they'll be very interesting. This one, I mean, this some of not all proteins are they give fluorescence. So what we do is you can take this one as a probe, as a phycocyanin, phycoerythrin, or allophycocyanin. You can use this one and for a tag this one and get the colors. So we also, that can be also used. And more than that, we use dessert milk product, ice creams. Now it also it been using for all this the food stuff also. So this is again same thing. Bacterial chlorophylls also will have a same kind of pigments. I don't want to talk about more on this one. Only thing, even colors, textile color also will be used, and a lot of ecological applications that we'll discuss. Okay, and as I said, specifically pigments and carotenoids, what it does, the same kind of functions. It has a more of the plant. If you take nowadays, I will discuss that one later. Astaxanthin. Have you heard astaxanthin? Astaxanthin is again a carotenoid pigment, where people are now working a lot on these pigments to understand. I mean, not understand, for industrial applications, not industrial, therapeutic application, but industrially they develop and they grow any cyanobacteria or algal samples and improve the astaxanthin. 10 mg is about 40,000 rupees, very expensive. Maybe we'll have one day tour Bionest upstairs, all of you. You can see that we'll have an industrial tour one day. This Saturday is packed, right? This Saturday is packed. We'll see that. Maybe this Saturday or 1 hour, we'll go there and you can understand much better. Okay, again, same kind of a lot of importance. It have more carotenoids, and with the whatever the plate, you will have more of, you live longer. Okay, so now all this happens because of the high yield of photosynthesis. So now, what are other importance with the algae? Of course, in the plant also you can convert in anything. If it is a biomass of plant or biomass of microalgae, you can get all these applications. So now, photosynthesis is not only give food, it gives many other elements. You see that valuable products, biofuel production. Valuable products means a small molecules where minerals, fertilizers, lipid fractions, lipid fraction will be used for many other things. So, you can, lipids also can be, whatever, the oils, you can convert an oils or, what we eat, lot of the oil stuff, antioxidant pigments, and there all be, what are the uses? These are the uses. Antioxidant pigments, vitamins, protein fractions, healthy supplement, and can be biogas, and carbohydrate fractions, health supplement, and they can also carbohydrates. The more the carbohydrates, especially plant, plant biomass, same thing, you put it as a plant, the biomass, you can get a nice, bioethanol production can be done. If you have wholesale biomass, they can make a health supplement. For example, I said wholesale means like, spirulina, a chlorella, even, the, some of them like, other pigments like a dunella, where Pavitra is here. Oh, okay. So dunella, or you can also make the tablets of the hematococcus. Several, there are several algal systems. You can make health supplements. So then, microalgae biorefinery is a very important nowadays. Is beyond photosynthesis. So people, to increase biomass, you need to have more photosynthetic performing algal system or plants, where once we have this one, so what do you do? Biochemical process. What if you do this process, take these microalgae and do the processing? Then processing is the one where you get fermentation, anaerobic and photobiological hydrogen production. Using that one, you get bioethanol, biogas, biohydrogen. So all these one are energy compounds. Bioethanol can be used as a fuel. Biogas, you can use fuel now for cooking. Biohydrogen now alternative fuels. It's called alternative fuels. You can replace the petrol where you can use biohydrogen can be used. The lot of research is going on to improve the the biogas, bioethanol, or biohydrogen, because the resources, natural resource of petrol, is going down. So the alternative source is only the biogas and biohydrogen. So if you go with the, these are the process. What are the process? If you go with the biochemical process, you get these products. If you get thermochemical process, more of heating energy. So that is a pyrolysis, gasification, liquefaction, compunction. So using that one, you can get all these gases, mostly gases. So trans-acidification, more of the lipids. If you have more lipids, so what you can get, you can get this one biodiesel, this trans-acidification. Mostly now, if you have more of fat, so where Jerome is here, Jerome Xavier, where is here? So what he is also doing on that same topic, to put more of lipids using CRISPR technology, and you get more of this trans-acidification, then improve these biodiesel products. So these are the some of the very important one. Based on this, a lot of people are now started their own companies, startups, and getting there are success stories, especially the we'll discuss about that a little bit later. Okay, this is the same. I don't want to go into this one. So this is one of the algae, that is a Hematococcus. Hematococcus is a green algae which produces high amount of the astaxanthin. I think we may have in the lab. Maybe you can see later. I think our Vijay is there. Vijay can show. Vijay Bunda, do we have Hematococcus plants? It's a reddish. Okay, so maybe he can show you during practicals time. So it will be like this, and it will be like this, and it will be like this. So I will show you one video where I'm advisor and also a kind of I don't say a partner, but I'm advisor there. This is our after my advice. Now they got all this very nice one. So initially we grow like this, the cells, it's a Hematococcus cells, it's a green algae. So, and once you grow green, what we do, we transfer to the ponds, mini ponds, here mini ponds. So, and then it goes to the bigger ponds. Now we'll see even much bigger. It will become high yield of photosynthesis. The photosynthesis will get yield, and when it get saturation of these supplements, the food supplement or a nutrient, then they will be convert into chlorophyll, convert into carotenoids, especially the astaxanthin. So this was Cherish, right? Yeah, Cherish, where we collectively have done this. It was in Medchel. It's an open pond system. So it's not only this Hematococcus, even Duinella. I think people are doing in Duinella, many other the algal system. So where okay, I will show you this life cycle. There will be so they have flagella. Looks green, greener. So they're moving nicely, almost converting to the red color. They'll be still alive. Even they contain more of carotenoids, they'll be still alive, the cells. So it happens cell division during the nutrients deficiency. Then they will become green again. When nutrient is there, they will become green. You just take them again, put it in the fresh media, they will become very green. I mean, this is a cell death. They can also happen cell death. For all these, photosynthesis is very important. Even if it converting carotenoids means there is a photosynthetic pigments play major role.
In the mature stage, when they have a lot of carotenoids, at that time, they go for photosynthesis or the external carbon source?
No, they do photosynthesis only. No, the nutrient you are talking, when it getting nutrient is less because less carbon is there. What it does, to protect themselves, cell and want to protect. So there is no, it's a kind of a senescence. It's leading to the senescence. So the senescence is the end of the, you say that end, but it is the beauty of this one. It can, when you give again, the cell division will be slow. Take them and put it in the green again, the regular nutrient, they will pop up immediately. Cell division happens, they will be one. But when the nutrient is, there is no CO2, there is no the ponds, yes, they can take CO2. Even if CO2 is there, they have cell senescence nutrient, other nutrients will be left. It's not only CO2, many other phosphate, it will become evaporated, right? So then they can convert into the pigments. It's clear? Okay, so this is again Hematococcus, and the Duinella salina is one where our Pavitra is working, but I don't know whether Pavitra has seen any time this color in your culture, right? Even in the end also. The cells have enough CO2. They are nutrient. That's why. But this also one of the company in the upstairs they are working, but now they moved to some other bigger space. It's also having beta carotene, high content of beta carotene, and especially the Hyderabad is very good environment to grow this Duinella because of it required dry culture, dry environment, and especially this Telangana region and the Rayalaseema region. You can grow nicely this one. Maybe Rajasthan, will be much better. So Israel is the one is a leader for this, because they have dry weather. They're growing a lot on this. These are all the ponds. I have visited these ponds in Israel. The same pond which I am showing here. Okay, so again, the same thing which I am showing you again and again. All these one you can use protein source, lipid source, carbohydrate source, vitamins, carotenoids, antioxidants. That's why I said those are Koreans and the Japanese. They live longer because of this. The pigmentation. They use pigmented food. So, and I wanted to tell you this one. Other importance with the using chloroplast genome is size is much bigger. You can do lot of therapeutic applications. You can, for example, anthraxine vaccine, several other therapeutic you can express and then purify it and can draw. The people are using chlorella, chlamydomonas, many other algal systems. For chlorella, and chlamydomonas, a big chloroplast, where they're using this kind of, you see in the cholera vaccines, any antigens, several things can be used chloroplast. That is another, apart from photosynthesis, they also use as well. We are also working on this topic. So where Yusuf is guiding one of the master students, trying to understand. So pigments have chlorophyll pigments and bacterial chlorophyll pigments, especially the carotenoid pigments. So when they absorb light, they emit, right? It has to emit. The mechanism here, I mean, these are the nanoparticles type. So if it is, let's say a cancerous cell, and then you go with your, the pigments tagged, or pigments can be tagged, or pigments can be supplemented. So you irradiate with the light. So what it does when it is irradiated, this produces oxygens, oxygen radicals, like singlet toxins, mostly singlet toxins, or it can also produce ROS, where the cancerous cells can be, this LED light, or you can also use, if you are using a bacterial chlorophyll absorb light in longer wavelength, and when they absorb light in longer wavelength, infrared regions, infrared is more efficient to kill the cells. So for you can say that the photosynthetic pigments are given nowadays through for the radiation, mostly radiation, and even chemo also will be now doing with this one, and so that will be a lot of the cell death can happen. It is happening actually. There are a lot of research, and especially Israel scientists, they invented this one. I thought they will get Nobel Prize, but somehow it is getting delayed for them. Wisman University, who invented, see that these are singlet toxins. It produces singlet toxins, and singlet toxin is again this radical. It can go and kill the cells, cancerous cells. Of course, there will be. That's why what they do, they probe, and wherever is the cancerous cells are there, especially they probe that one, tag that one, so that it will be killing only those cancerous cells. This is very interesting and very important. We also have to do this research in the lab. So same thing, photodynamic therapy. You can also see that chlorophyll. This chlorophyll, so it produces ROS, and in the cell, it kills all these. Apoptosis can happen. Then further cell death can happen. Through that, you can get a lot of the medicinal use or medical use or this is the same thing. The photodynamic therapy. You can see that wavelength. They use specific wavelength because the pigments have chlorophyll A, chlorophyll B, chlorophyll C, or carotenoids, several other pigments, bacterial chlorophyll. They have different wavelength it absorbs, so that you can use various light to kill the cancerous cells. So even say that body by kidney 24 to 70 hours. So but it remains in cancer cell for a longer period of time when the photosensitizer cancer cell exposed to specific wavelength and produces abundance of oxygen that builds and kills the cancerous cells. So this is what now in the practice, and these are the apart from your food and other things, the therapeutic importance is also there. Okay, so now, so this is about the application part. There are people are now working a lot on the applications to understand and characterize and go for industrial application through photosynthesis. So, and I want to ask one question. If let's take, maybe I don't want to ask question, but I will tell you, if you increase photosynthesis, there will be a lot of other problems. What we do, there natural strains. Always we take natural strains for such kind of one. But you can also manipulate inside. Let's say I want to have lipids more, you can do it. I want to have pigments cannot be more. You cannot produce pigments because if you take pigments more, it may have others lead, it can lead, but it can synthesize. If you change something, pigments can be changed, but pigments itself cannot be improved in our research. But let's say there also like a Rubisco, for example. You cannot play with the Rubisco. If you disturb Rubisco, the entire system will collapse. So Rubisco, if we express, because it is it can produce a lot of other reactive oxygen species and can kill the cells also. Anyway, so now just want to, these figures might have showed already by many people. So now the pigments, since the topic is the pigments, where the pigment stays here. So this is photosystem 2 and the cytochrome B6F complex, photosystem 1 and ATP synthase. And when you have a green here, these are light arresting complexes. All together, photosystem 1 and photosystem 2. This is about the photosystem 1 have more pigment than photosystem 2. And this about 200 plus pigments will be there. 200 plus means 220, depending on species, about 218 to 20 is there. Going to this one, this have about 240 to 50 chlorophylls of photosystem 1. These two will have a pigments, high abundance of chlorophyll A, chlorophyll B and carotenoids. So the chlorophylls will be mostly present in light arresting complexes, as well the reaction center. The reaction centers, I think they have explained, right? HRO has explained. These two will have the pigments, so which are very important to harvest the light. It can be, chlorophyll A is the ultimate, they have to harvest, and whatever, the splitting process happens. So now, why pigments are required again in photosystem 1? Can any of you tell why the pigments are required again in photosystem 1? Anyway, this is a linear electron transport, it goes. Why they have to have again pigments here? Anyway, so it's like a, if you like, if you just, and I don't have chalk piece here. If you throw like this, it goes whatever, from steady state to the excited state. So then it has to fall down. So what happens here, when it through all this linear electron transport up to here and comes here, and all this electron transport, proton motive force, they lose energy, right? When you are running marathon, any of you been marathon? How many of you there marathon? 1, 2, 3, only 4, 5, yeah. So let's say 10 kilometer, 10K or 40K. 40K have been anywhere? 10K? Okay, 10K means 10 kilometers. So when you are walking or running, you will be exhausted. So when you exhaust, what they do? The guys will be there, they will feed you some juice or some energy drinks. So now the same thing, that means you are tired, you are losing energy. Similarly, you are losing energy here. By the time it comes to the before photosystem, I am tired, I want something. So what it does, you have to have energy again, solar energy, to pump these electrons to the linear transport. So that's why the pigments again mimicking to this one, but you don't need that much energy. The one you have ate in, let's say, a morning, it produce more, but in between you need a small, you need to have a little energy. That's why they have a longer pigmentation. They pick up, then transfer the energy to reduce NADP to NADPH, NADP to NADH. So, and here also core proteins and lighter, both have pigments, but light arresting complex have more pigments. And there are different kind of, there are a lot of things you can discuss. Each pigment have a different kind of, the energy, how they transfer, because so many pigments, it's like a network, how they'll be connected, dotted, they finally enter into the reaction center and you get. Okay, this is beautiful. I always tell my students, you have phycobilisomes mimicking same photosystem thylakoid complexes. This is a thylakoid membrane and sitting on the phycobilisomes, phycocyanin, phycoerythrin, allophycocyanin. These three pigments, different colors, and absorb the light and transfer to the photosystems. The rest will be almost same. So very interestingly, this is like a light arresting complex. Replaced, I know in the higher plants they have a clear light arresting complexes. They are subunits, but those light arresting complexes are replaced by phycobilisomes in cyanobacteria. And the reaction center they have a green again pigments. And interestingly, there won't be light arresting complex in cyanobacteria because it stays in the water. They don't require that much of energy. So whatever the energy coming from reaction center that have pigments, that solar energy is enough to transfer this one to linear electron transfer. So these lakes are beautiful because of the cyanobacteria. It's a reflection where a Niagara or wherever you see the some beautiful when it sun shines and the pigments will be there. That means cyanobacteria or red algae or something, and they will be looking like that. Wherever you see that beautiful because of the algae or cyanobacteria. And bacterial chlorophylls having abundance of chlorophylls, rings like structures. They also have observe longer wavelength and then transfer the energy, the Q cycle, which is more of make into cyclic electron transport. So plants and algae and cyanobacteria, they have a cyclic and non-cyclic, but in bacterial system, they have only cyclic electron transport. I think Professor Raghavendra has discussed, where they will have they don't require that much energy. They don't produce NADPH. So it is only ATP it produces. That's why it have even in cyclic electron transport in plants, they have only ATP. Through cyclic electron transport, you get only ATP. That energy is enough to survive all the bacterial photosynthesis. It's mostly in anaerobic photosynthesis. This system will be happy. Okay, I think I will be at the end. This I think I'm not going to discuss because it's already HRO is discussing all this one. He is going to discuss all. So thank you, and you have any interactions? Not a question hours, but interaction. If you have, we can have any doubts, we can clear.
So, in the photodynamic therapy for cancer, usually ROS can diffuse out of these cancer cells as well. So it can go to the normal cells as well. So how this limitation?
That's what I said. They have to now the technology has developed. So you have probe. The cancer cells should be tagged with the pigment, and through that you are targeting directly to the cancer cells.
Yeah, after that the cell death will happen.
It may be there, yeah. Always side effects will be there, but it's more efficient. If it is probed with let's say even if you are giving chemo or something, the bacterial chlorophyll will be tagged, and these whatever the location you are in it, let's say a tumor is there, you target only tumor. Yeah, cell diffusion will be happening, but the other cells can be come back again, right? They can grow again, even if it is that some side effects are there. But the cancer is always side effect. But the mechanism is the one, the ROS generation that kills the cancerous cells. But what mostly they probing, they tag, not probing, tagging with probing exactly to the inside the cell. So that's what the therapy. So you don't have questions. At least you can start your company. You see, like a lot of pigments. You can have people are now there earlier. Spirulina now was the small scale industry. People are growing at home, backyard, and that, there are a lot of in the villages, many of them still now do all these clothes or whatever, then they collect industry people. Similarly, spirulina was, backyard, the people, a pond, they grow just a simple water, you grow it and filter it, and, you dry the powder, and, the company people will come and, collect the pigments. That's what's been there for now, because now the industry itself, they started their companies, and, they're now producing more and more. So with the small scale, it's not, but still people are doing, specifically Mysore region. So there are a lot of people are now aware, they're in the backyard, they're growing and getting like a mushroom culture. You can do it at home. This algal culture also can be, you can grow, and you have to standardize. They will go and, if you go to CFTRA, have you heard CFTRA Institute? Food Institute, CFTRA in Mysore. So they will also give training on this algal culture, how to grow and what kind of conditions. Then you can implement at your home and get some money. I mean, precautions, there will be contamination can happen, other bacteria can grow, that we have to be careful. But, before harvesting, you have to check. Sometimes, toxic bacteria can grow, but we have to be very careful during that time. But, before, it's going to, the feed or whatever. And also, I forgot to tell you, this pigment, all this one also used for animal feed and biofertilizers. Nowadays, you can, algal culture can be, the waste of algal culture also used for biofertilizers. Yeah, there will be contamination that we should be careful. But in time to time, you have to see, before harvesting, you have to see under microscope. And if it is apart from your spirulina or something, you have to throw or see that what is the condition. But most of the algals, they are safe. It's edible. They're all edible. NADPH, bacterial photosynthesis, anaerobic photosynthesis, because there is no CO2, right? So yeah. No, this will be used for other, the production of, all these molecules will be there. The ATP, ATP will be utilized again on the different cycle, not Kelvin-Benson cycle. So it will have to be, synthesized. Carbohydrate will synthesize, proteins, lipids, all of them will be synthesized. And most of the, the bacterial, they will be using this one as a bioremediation, all these bacteria. Okay, it looks like no interaction, but you might have understood everything or maybe not. It's simple, I have not complicated story I didn't tell. It's more reachable and can be understandable. All of you can understand. And as I said, one of the day, you can, all of you, that 15, 16 students, will visit the industry by upstairs. We have the, whatever, you're having food, that all industry, all, even there is one company working on leaf protein, Rubisco protein. So they leave from waste leaves, they isolate Rubisco, and Rubisco is again, rich of protein, abundance of, highly, abundant protein, where it can be used for a supplement of, or you can say vegan protein, veg protein, where protein replacing meat. They are working on those aspects. We can visit. There is some plant tissue culture related to plant, but many other, there are a lot of enzyme and biosimilars. You can go and visit the company. I will arrange this Saturday, or depending on who is our Your room is there. What is the program? Can you see program? It will take time. But anyway, one of the day you will be going there. Okay, then if there are no questions, anyway, now I'll be continuing other two talks. You can also discuss any question if you have any question. Thank you.
So I talked about the structure of PS1, LH1, super complex. Something like this. And then in the last, I just want to show how the pigments, chlorophylls, carotenoids, are organized in the super complex. So this is a view from the stromal side, and these are transmembrane helices. No, these are transmembrane helices, something like this. And this is a PS1 core, okay, reaction center. And we have talked about photosystem too, but this is PS2 core. PS2 core is made of mainly 4 subunits. Reaction center, I should say, yeah, core. And the PS1 made of mainly 2 large subunits. So the size is different, okay? This is PSAA is about 7 or 80 kilodalton. PSAB is the same size. And PS2 is reaction center is PSAAD heterodimer, and core antenna PSAC and PSAB are present. That is core antenna. So you may think that the size and also the primary sequence of the amino acid sequence of PSA B and PSAB C, PSAD B, PS2 and PS1 amino acid sequence very different. It's not very much conserved. But if you look at the arrangement of the transmembrane helices, okay, of PS1 and PS2 are very similar. PS1, PSAA, if you look at PSAA here, it's here. It's N-terminal region, this domain, the 2 domains. N-terminal domain is here. C-terminal domain, C-terminal domain is here. Okay? And also very symmetrically, PSAB is the same. N-terminal domain is here, and C-terminal domain is here. And N-terminal domain contains 6 transmembrane helices. And 2 pair A, B helices, C, D helices, E, F helices have pair. Okay? 3 pair of alpha helix is N-terminal domain. The PSAB N-terminal domain also the same. AB, CD, EF domain, alpha helix. And it's a kind of circular structure. And in the C-terminal domain, 5 helices are present. And 1, 2, 3, 4, 5. And also PSAB, also C-terminal domain is 5 helices. And also, if you look at carefully, the arrangement of this alpha helices is like something like this, here and here, no? Like shaking hand. Okay. If you look at the PS2 core, this is reaction center. AD, this is heterodimer, also very similar each other. and 5 helices. And also arrangement of 5 helices like this and like this. So PS1 reaction center component is present here, and PS2 is present here. So these two domain looks similar. And this is a terminal domain of PSA A and PSA B. It's associate antenna, only antenna pigments. So we call this part is core antenna. This part is reaction center. Okay, core antenna, reaction center. And PS2, it's different on the different polypeptide, PSA C and B, core antenna are present. But this arrangement of alpha helix, the pair, 3 pair of alpha helix are circular, like this. So it's similar, this part. Okay, and this contain only core antenna and the reaction center here. So PS1 and PS2, because in the beginning when we determined the amino acid sequence of PS1 subunit and PS2 subunit, they were not similar. So most researchers thought photosystem 1 and photosystem 2 are developed independently from the different origin. But if you look at this 3-dimensional structure, primary structure it's difficult to say PS1 and PS2 are similar. But the 3-dimensional structure are very similar each other. And then specific, some very important amino acid that is involved in associating a pigment or cofactors are conserved. So now we think that the PS1 and the PS2 are evolved from the original, the same ancestral reaction center. But in here, PSA is separated into 2 subunits, and the PSAB was separated. Or I don't know, we don't know, these 2 subunits were fused to a single polypeptide. That happened. And if you look at the number of pigments, PS1, PS2, more chlorophyll molecules you can find in PS1, right? The PS2, no, more spaces. That's different. But PS2 has contain more a lot of light harvesting complex 2, turmeric LHC2 outside the this region is surrounded by turmeric LHC2. So total antenna pigments number is more. You can find more antenna pigments in PS2 unit. Okay. Okay. Another interesting thing is if you look at the structure from the side, the thylakoid membrane, the section, and this is the position of the chlorophylls. This is reaction center. Okay, PS2. This is reaction center, and this is a core antenna, and this is reaction center, and this is core antenna. Okay, so as I told, if you remember, LHC2, in LHC2, the structure I told that the pigments, chlorophylls are distributed. Appear to be distributed more evenly But if you look at the section of the structure, the chlorophyll is concentrated on stroma side and luminar side, but less chlorophylls in the center. And in the case of the PS1 and the PS2 core, no, this is not a light harvesting. From the stroma side, the pigments are distributed evenly, but if you look at from the side, stroma side, luminar side, pigments are more concentrated, right? In the middle, you don't see many pigments. That is the case in PS2, no? Stroma side, luminar side, and stroma side. More chlorophylls, and in the center, less chlorophyll. Of course, in the reaction center, here, the photochemical reaction occurs. So electron is transferred from the luminar side to stroma side. So the redox component, including chlorophylls, are located across from the luminar side to stroma side. So this is exceptional, right? That is the case. And the photons, for example, PS2 photons, when the light photon absorbed by chlorophylls here or here, and then chlorophyll becomes excited. And this is excited state is transferred among the chlorophylls, and then finally reach to reaction center. Okay, so probably we think that from the structure, these chlorophylls are closer to the reaction center. So most of excitation energy is first transferred to here, no? And then finally transferred to the reaction center. In the case of PS1, there are some chlorophylls here, but finally, this is more chlorophylls. But the excitation energy transferred from here to reaction center. And one chlorophyll like this is more the closest. Not every chlorophyll, but some specific, two specific chlorophylls are close to the reaction center. So it transfers the excitation energy. That is we can speculate these kind of things from the structure. So this high resolution structure helps us understand how the excitation energy from the antenna to reaction center are transferred. That is the case. So as I told before, even equal distribution, instead of installation concentration, the luminar astronomer side is make the distance between the chlorophylls shorter. That is very important for efficient excitation energy transfer from chlorophyll to chlorophyll. No, so that is a structure analysis made increased our understanding of the structure function of these pigments. Okay, this is a structure. So conclusion is that this very organized, precisely organized arrangement of pigments are made possible by protein, no? Scaffold. Proteins arrange and fix these pigments at proper position. So this is precisely done. So there are many subunits are present. So next topic is how this PS1 complex is made. So particularly the many component, particularly I'm going to focus on only subunits. Many subunits are synthesized independently by translation and then come together, not randomly. How it happens. So this is we call assembly process. Okay, so I want to change the slide to another file. How could someone help me to change? Escape, and then close this, and then lecture. This one. Okay. So this is next is assembly process. Is that okay? Okay. I took from my presentation from some conference, so just slightly different. I show you the highest mountain in Japan, the volcano, Mount Fuji. It's summer, so it's not covered with snow. So, but it's very our symbolistic mountain. And okay, as we discussed, this is a PS1 LH1 supercomplex of chlamydomonas. And this subunits composition. So in chlamydomonas, PSA, B, C, J are encoded by chloroplast, and the others are encoded by nuclear genome. This is an example of the component composition of vascular plants. This is chlamydomonas and cyanobacteria. This is a prokaryotic. So in the case of chlamydomonas, these are chloroplast and others are nuclear encoded. And the presence of subunits, vascular plants and chlamydomonas, if you compare, there's no PSAM is missing, but in the cyanobacteria it's present. N or P is not found in chlamydomonas, but present in vascular plants, not in cyanobacteria. PSAX is present only in cyanobacteria. PSAGH are present in the vascular plants and chlamydomonas, but it's lacking, deficient in cyanobacteria. So there is some difference in peripheral subunit, but important subunit, PSA A, B, C is associate redox cofactor. So these are all conserved among the cyanobacteria, green algae, vascular plant. And LSC1 is vascular plant has 4 LSC1, as I told you before. And sometimes in some other NAD, PS1 NADH supercomplex, LHC5 or 6 transiently minor, as a minor component is present. But it's usually PS1 contain 4 LHC1. In chlamydomonas, 9 different LHC1 and total 10 copies. The cyanobacteria has no LHC. It contains phycobilisomes. So instead of LHC. So PS1 also accept the excitation energy from phycobilisome that is different from type of the phycobilisome of PS2. So this is different part, peripheral. So but this structure must be assembled properly to express the function of PS1. So this is how PS1 complex is assembled. So if you think about the motor car assembly factory, car is made of, I don't know, 10,000 or many parts, right? But if you just mix these parts and then shake, but you don't find the car, not functional. It's just a mixture of parts, right? So in the factory, what happened is, can you see this? The robot assembling part at the proper position, right? It's not randomly. So and also the order of assembly of parts are fixed, right? If you put the parts in the wrong steps, it's difficult to make cars. Okay, so like like a robot or workers assemble motor car step by step, and then gradually cars are assembled. Okay, and in the final, if you purchase car, of course you don't find in the car the workers, factory workers, or a robot to assemble. No, only you can buy car. It is a very similar step of assembly of complex. There are many components for example, in the PS1 complexes. So probably each subunit assembled into to make a final complex step by step, probably. Okay, from the center to the peripheral parts, probably. No, assembled step by step. We don't know how many steps there are, but and also like factory workers or robot, each parts or each subunit, when they integrate it into the complex, there must be factors that assist assembly. That is called, we call the assembly factors. That is temporarily involved in this process. And when mature PS1 complex was assembled, this factor disappear because we don't find. So to understand how the PS1 complex is assembled, we want to know these assembly steps. And also, we want to find assembly factors. Okay. So concerning assembly factors, this is a Chloroplast DNA of Chlamydomonas. And we had by chance analyzed two open reading frames. Open reading frame means there is a initiation codon, methionine, and then stop codon. But we don't know if this is expressed, transcribed, translated, and we don't know function. So in that case, we called named Chloroplast hypothetical chloroplast open reading frame. So hypothetical means probably this may be important, may have a function, because in this case, this hypothetical chloroplast open reading frame is conserved in plants, chloroplast, and even cyanobacteria genome. Okay, very conserved. So that must be has function or expressed. So there's a numbering from the large size. For example, it's YCF1, 2, and YCF3 and 4 are present here in chlamydomonas. So hypothetical chloroplast open reading frame 3 and 4. So we by chance knocked out these two genes independently to see a phenotype. Maybe the strategy to do a chloroplast transformation will be I will talk in the other lectures. So we knock out anyway. Then we analyzed the phenotype. Then we found PS1 is gone. Although YCS3 and YCS4 are obviously not a component of PS1, right? But these two are important for accumulation of PS1. So we thought, oh, this may be a factor that assisting or involved in somehow PS1 synthesis. But this was not reported before, so we don't know how this true. Actually, we found that the YCS3 and YCS4 are translated, and proteins present we detected by immunoblotting. We raised the antibody against the YCS3 and YCS4 and found it's present in the cells, in the chloroplast, in the thylakoid. And if we knock out this, of course, YCF3, but YCF4 is present, PS1 is gone. And YCF3 present, YCF4 knockout, but PS1 is gone. So that's happened. So then we thought that's how YCF3 and YCF4 are involved in PS1 accumulation. Probably we thought at the time already that maybe important for assembly of PS1 complex. But we needed the evidence to show this. So what we did was affinity purification of YCF3. As I discussed before about the chloroplast SRP 43 and RB3.1 affinity purification and find out interacting protein. So this is a method. Now, first we transform the chloroplast gene of YCF3 or YCF4 and then HA tagged genes introduced. Okay, and then check that even infusing HA tag at either N terminal or C terminal still works because PS1 is assembled. In the case of chloroplast gene, we don't need to worry about N or C terminal side. We can insert N terminal side. So we inserted either N or C and to see which is better. And then we purify this. HA tag is the amino acid is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 9 amino acid. And so we put into the spin column, not small column in the beads that specifically bind the HA tag sequence. So in the mixture of the protein from the cells, HA tagged proteins and the cross is cellular proteins, the mixture, and then we add the beads, no, the antibody against HA tag, mixed it, and then this part is put into a little bit small or larger tube, and then spin down. Okay, so cross is a cellular protein without HA tag is eluted out, and beads associating HA tag protein remain here. Is that clear? You can see. So if you do this washing few times, then you can completely remove the protein without HA tag. And then peptide solution containing this peptide, synthetic peptide. Then HA tagged protein is eluted out. And then these beads associate this synthetic peptide, because concentration of synthetic peptide is much, much or higher, right? So you can replace this. Then you can purify this HA tagged protein. Okay. So YCF3 HA affinity purification. This is the result. We use wild type as a negative control. You don't see practically nothing. But there may be some band here. But YCF3HA, you can purify here. And a little bit smaller contamination, no similar contamination. And also, there's another band here. Okay, so pure, I should say, because the amount of YCF3 is catalytic amount. Okay, so even though we can purify very nice band here, and then may be minor, but if you look at the intensity, compare the intensity, it's almost no contamination, practically no contamination. Concerning this interacting protein, some German group, Arbas, also purified using a tobacco. Their result is here. This is negative control. They use a flag tag. So even negative control, there are many proteins like here. And this is positive control. Positive, this is the YCF3 flag. So YCF3 was here. And others are, no, these are all non-specific contamination. And then they also find a specific protein that is lacking in the negative control, but here. So they named it YCF3 interacting protein 1, Y3IP1. But we found this, also did this experiment with later, published 8 years later, but our preparation is much cleaner. This was done in the Nerpalis leader from this Hyderabad. He was a postdoc in my laboratory, and then he did this experiment. Huh? Srida. So what is nice compared to this? Purity is high. It's just looks nice. Not only looks nice, but it's very useful. If you detect it, the major protein is these two, almost stoichiometrical amount. But if you detect it, the immunoblotting, this is Y type. You don't see any. But this YCF3 tagged purification, of course, YCF3 here, tagged, and Y3IP1, and a little bit YCF4. You don't see here the band, but it's what's present. And what is important is small amount of PSA A, F, but not other. PS1 subunit was small amount of PS1 subunit was there. But if you use this kind of not so pure preparation, if you see polypeptide other than YCF3, nobody believes that that is authentic interacting protein, but it's maybe contamination. Okay, so since YCF3 is essential for PS1 accumulation, we expect it's possible that the YCF3 has somehow had interaction with PS1 subunit to assist PS1 accumulation. That is, to assist the PS1 assembly. So to assist assembly, the factor must somehow interact with PS1. As a motor car, as a car assembly factory, robot catch one parts and put into the car. So there's interaction, right? The same thing happened. So this immunoblotting data suggests this interaction. But of course, there's a possibility that this interaction with PS1 is non-specific contamination, non-specific interaction. Okay? So what we did was radio labeling experiment. I told you before that newly synthesized proteins, after pulse labeling, newly synthesized proteins are labeled. Okay? After chase, pulse and chase experiment, then if the interaction is stable, a labeled protein stays there. But interaction is transient, radio-labeled protein disappear. Okay, so we checked. We labeled protein with S35. Oh, sulfur is better than carbon-14 to detect, more sensitive. So this is a thyroid membrane. Pulse labeling, C is chase. Pulse labeling, short minutes, 1 or 5 or 10 minutes. Chase is 1 hour or 2 hours, something like that. Okay. In the thyroid membrane, you see a labeled protein like this. And after pulse and chase, you don't see much difference in labeling. See, that means in the thyroid membrane, the pulse labeled protein is present, and also even after 2 hours, this protein stay in the thyroid membrane, as expected, because synthesized protein is present in thyroid membrane, not after its synthesis. That is as expected, no, nothing special. So it's actually this ATP synthase alpha and beta subunit, and this I don't know exactly, but this is probably LSC proteins. Okay, because this time pulse labeling was not 1 minute; it's a little bit longer. So LSC is proteins labeled. Okay, from this pulse labeled and the chase labeled, pulse labeled chase thylakoid membrane, we purified YCF3 the same way, but using labeled thylakoid. Then what happened? Okay, so YCF3 HA tag here, the PSAF here, PSAF was a little bit present. You can detect, okay? And also there's many diffuse band, but YCF3, YCIP1 was somewhere like here, okay? According to the position. And some proteins are there. But what is important is PS1 reaction center proteins, PSA A and B, labeled proteins were present. Okay, reaction center protein. We couldn't detect by staining because there are some diffuse band around here, and also amount is so low. But label was clearly shown. And after chase, reaction center protein, this diffuse band, disappeared. And also PSAF was also disappeared. Somehow, YCF3 also labeled YCF3 disappeared. Y3IP1 present. The others are also similarly present. So this means that the PSA A and B newly synthesized PSA A and B are transiently bound to YCF3. And also YCF3 itself turnover. This means turnover. This is newly synthesized and it's disappeared, digested. But Y3IP1 is stable. Not much labeled, but it stays. The stoichiometrical amount, but this turnover is fast and this is slow. But still, they present as dimer or oligomer, but stoichiometrical amount according to the staining band. So, so this data clearly show that YCF3 is associated with newly synthesized PS1 protein. So which this result is consistent with YCF3 its assembly factor, because assembly factor interact with newly synthesized PS1 protein. Okay? So this data is clearly show that YCF3 is one of the assembly factor of PS1. Okay. But this is more related to only reaction center protein, or PSAF a little bit, but mostly, but other PS1 subunit were not interacting. So from this data, we conclude that mainly PSA A and B, assembly of PSA A and B reaction center of PS1 is more core part. It's an important part. In this assembly, YCS3 is involved. So this is the very initial steps of PS1 complex assembly. And also Y3IP1, YCF3 interacting protein, what this does. So Y3IP1 minus mutant, also in the chlamydomas CGL51. CGL1 means chloroplast green lineage, so it's related to some photosynthesis or something. Number 59. This is another name of Y3IP1. So we obtain the CLIP library of Chlamydomonas center, so we can obtain by paying $100 US dollar. So we can get this. This delta Y3IP1 mutant. Okay, the YCL3 mutant, YCL4 mutant, we check the growth of the cell. This is a dark condition, but on the top media. Top media contain taurus acetate phosphate. It contains acetate. That is a carbon source for respiration. So chlamydia cell can grow heterotropically under this condition in the dark. So these 4 strains grow well. Okay, and if you illuminate here 50 micromole photons per square meter per second, wild type grow better. But YCL3, YCL4 very photosensitive, so it didn't grow. Y3IP1 a little bit grow, very much reduced, but it's still better than YCL3 and YCL4. And this is a HSM media, minimal high salt minimum. This lacks acetate. Okay, so cell can grow only under the photoautotropic condition. So Y3IP1, YCL3, YCL4 didn't grow at all. Okay. So Y3IP1 is also very important for photosynthesis, probably PS1. And we also checked these 3 strains by immunoblotting the presence of related proteins. This is wild type, controlled wild type. We loaded 100%, 30, 10% of the sample. This is a dilution series. And also PSAA, YCL1, YCL3, YCL4, you don't see the signal, but very, very slightly delta Y3IP1 mutant accumulate PS1. Okay. You can see it's less than 10%, maybe 5%, something like that. And also Y3IP1 present, but this mutant lacks, of course. YCL3 mutant lacks, but Y3IP1 still accumulate YCL3. YCL4 mutants accumulate YCL4. YCL4 mutant lacks YCL4, but others are still present. So, because we detected very, very small amount of PS1, and also photosensitivity is a little bit less than delta YCL3 and YCL4. So we expected that there may be small amount of PS1 is assembled. So we isolated thyroid membrane, solubilized with mild detergent, and separated extract by circumstantial gradient, as shown many times. This is the top of the gradient and the bottom of gradient. It's for Y type. We can see LHC2, PS2, PS1 LHC super complex. Nice 3 green band. And from YCL3 mutant lacks, that which lacks PS1, it's only LHC2 and the PS2 and the LHC1. Without PS1, LHC1 accumulates, so 2 green band. From the delta Y3IP1, that accumulate a small amount of PS1, 2 band, but slightly green band are present in PS1 and LH1 super complex. The presence of small amount of PSA1 in this mutant mean that it's assembled. not the protein presence of protein, but it's assembled. So we took this fraction also from wild type and from this mutant. Also we checked blotting of this fraction and checked PSA, C, D, L, LSC4, LSC. So the PS1 and LSC1 protein was detected by blotting. Although the amount is lower, but still present, it seems that its assembly is almost perfectly worked. Then we isolated this fraction. We measured PS1 activity. Okay, so the wild type show when you illuminate P700 or PS1 reaction center oxidized, and then you stop the illumination, it's reduced. So it automatically. And delta Y3IP1, this is small amount of PS1, so it's also show activity. So it's assembled. So I thought I mentioned that the Y3IP1 is important for PS1 accumulation, but this shows that it's not essential. Without Y3IP1, PS1 is assembled in a very inefficient manner, but still possible. So we said, because Y3 is present already here. So what happened? So essential component is Y3.
Sorry to disturb you. It's not related to the topic we are talking, but I have a doubt in the Western blot. You have
Sure.
Western blot in the second figure. Yeah, actually for mutant confirmation, no, the top right side, second figure, Western blot.
This one?
Immunoblot, yeah. That figure for mutants, exactly we cannot see the respective bands, right? YCF3.
Y3.
Yeah, mutants we are not able to see. Respective mutants we are not
This
getting
one?
the
These
bands.
two.
No. I mean
one?
to say the respective mutants. Let's take Y3IP1 is a mutant, right? Yeah, in that the expected protein signal was not there, right? So this is antibody generated specifically by you, or it's like commercially available antibody
You
used?
mean this one?
Yeah, down. I mean the Y3IP1 is there, right? The second line, yeah. In the mutant there was no band. That's
No, but
right.
of course,
That's correct.
This is mutant.
Yeah, but the antibody is specifically generated, or it's like commercially available antibody?
This is specifically generated from the by overexpression of E. coli.
Okay, yeah. Otherwise, again, non-specific. They may bind somewhere else also, no?
This is a specific.
Because I do have the mutant. So when I thought of doing Western blot, this question came. So if somewhere else they would have designed the antigen, they may still bind, even though if the insertion is there, still they could signal.
You mean the small truncated proteins?
Still they give a signal. Yeah. But I think you should
but anyway, we couldn't detect anywhere. Of course, there's the possibility small amount, but it's maybe there's small possibility, but
yeah, because I want to do that. So I had a doubt whether it should be specific. Then I should design the antibody like you specifically. Then only I can get the expected results.
This was probably, I don't remember, it's a synthetic peptide or overexpression. Probably this is synthetic peptide.
Okay, yeah.
So, of course.
It should be specific to the site of insertion. Then only the cleavage, it will disturb the sequence. So we may not be able to get the...
In the middle of the coding sequence,
yeah.
The selective marker is inserted. So usually protein will not be expressed. This possibility is, I don't know, we thought it's unlikely. Of course, in the 100%, but 99% unlikely. Okay, that's it's always good to suspect data or possibility. So now the question is, this is knockout mutant, but in the part of the Y3IP1 may be expressed and translated and present in somewhere in a small amount, but that couldn't detect by immunoblotting, and that also had assembly of PS1. But yeah, when we submitted paper, no reviewer complained about this. So we just possibility. Yeah, okay. Always this kind of question is important to ask. So Y3IP1 and YCF3, this is additional YCF4, but we could detect YCF4, but this comes and then assembly occurs. And this YCF3 turnover the same rate as PS1, so that means we thought it's unlikely, but the YCF3 is once it's assembled PS1 one time, it disappears, and new YCF3 is synthesized. That is a waste of energy, but it's according to the pulse chase labeling experiment, that may happen. So, but we don't know why this is important, this turnover. This is unexpected data. This is until 2. Oh, finishing? Oh, finishing.
We can wait for.
Okay, so I will stop. I will talk this to be continued tomorrow. Okay. Any question? Other question? Sorry for. There are so many detailed data, so it takes time to explain very well. So, is there any? Yeah. Could you use a microphone?
Okay, my question is from the first part. PS1 core has more pigment molecules, you showed, right? PS1 core, more pigment molecules, more chlorophyll molecules arranged.
Yeah, a structure.
So when photoinhibition happens in highlight stress or something, it affects more PS2.
Turnover is PS2, yeah.
PS2. So if more excited chlorophyll molecules, there must be chance for more ROS production in PS1. So is that this packed arrangement of pigment molecules is helping its efficient energy transfer, and so that the ROS production is not happening, or why it is like more protected than PS2?
I think PS2. Okay, it's a question. It's a PS. There's the two photosystem, PS1 and PS2, and under the highlight condition, PS2 is preferentially photo damaged by, and then that is photosystem 2 has the system to repair the damaged PS2. Then once the stressful light is removed, it's very quickly PS2 is recovered, right? The PS1 has no such a system. So once PS1 is damaged, there's no repairing system. So the PS1 must be synthesized. Whole complex must be synthesized. So it takes time, and the photo damage of PS1 is affected very more significantly. So basically PS2 photo damage is important for protecting PS1, right? PS2 supply electrons to PS1. So, but under strong light, if PS1 works, it generates a lot of reactive oxygen species. So if PS2 is the damaged, the electron will not be provided to PS1. So without electron, PS1 cannot maybe generate much less reactive oxygen species. Okay. So to avoid photo damage of PS1, donor side of PS1 must be oxidized. So that means that the reducing side of PS1 is reduced, over-reduced. That generate reactive oxygen species. So to avoid this, there's a cyclic electron transfer around PS1 to avoid the over-reduction of reducing side of PS1. And also PS1, PS2 is blocked, then electron will not come to PS1, so PS1 cannot work without the electron. And also, if the luminal side is over-reduced, acidified, the luminal side acidified, so then photosynthetic control occurs. That is, I haven't mentioned this. The strong acidic condition, electron transfer from cytochrome B6F to PS1 is suppressed. But there are many safety valves for not producing reactive oxygen by PS1, I think.
And also, you think this pigment molecules arrangement is making it more efficient for the excitation energy transfer in PS1?
In PS1, yeah, more than PS2. I don't know. That is both are very efficient, but PS2 quantum efficiency is lower than PS1. So PS1 is very efficient. Quantum efficiency is almost 1.
Yes.
So the PS1 in synthesis is how to say, it's not involved in photo inhibition compared to PS2. So this is a very important point. Is that right? Right, correct answer? My answer is
Yes.
okay.
Thank you.
Any question? No, don't hesitate to ask me anything. That if you have a misunderstanding, that is very good to understand better, and also maybe others also the same question. So it's good to have give questions and the answer that helps understanding a lot. But maybe later. So maybe we are hungry. Okay, thank you. So we continue tomorrow.
Measure? No. No, not exactly direct reaction process. So is it a direct measurement or an indirect measurement? Why the name is there infrared? Why not anything else? So first of all, it will only analyze your gas, okay, and that to be all heteroatomic gas molecules. Heteroatomic gases molecules means homoatomic means oxygen, okay. Heteroatomic will be your CO2, methane, everything for that are different atoms, that is heteroatomic. So it will never analyze your oxygen or anything. Got it? And another thing is that all the heteroatomic, they absorb infrared radiation. You got it? After the absorption of infrared radiation, each molecule will, suppose 10 molecules are there, and they will absorb, for example, I'm talking about 5 to 7 infrared radiation they will absorb. Now, if the molecules get increased to 20, so that absorption will be directly proportional to the infrared absorption, means the molecule number. So if it is 10, then for example infrared absorption will be 10. If it is 20, then infrared absorption will be 20. So that way the machine measures whether the CO2 is utilized or it is less utilized. Obviously, if a plant is under photosynthesis, so CO2 will diffuse through stomata into the, if you are providing actinic light, then the CO2 will be fixed. It will enter through bicarbonate. It will first enter into the stomata, then convert into bicarbonate, then through bicarbonate it will be fixed through C3 cycle or dark reaction. So that dark reaction, Rubisco activity plays an important role. Okay, so this does not exactly measure the oxygen evolution or anything, because this already I told you, this is a only measurement for heteroatomic molecules, heteroatomic gases, not all molecules, heteroatomic gases. So it will measure only the CO2 concentration. So CO2 concentration, it will see if a plant is performing well, the CO2 absorption will be more or less. Then with the CO2 absorption, it will link all other things. Stomatal conductance, if a CO2 is more absorbed, then it will calculate its stomatal conductance based on its internal calculation parameters. Then comes your transpiration rate. Then from transpiration rate, you can go for net CO2 assimilation. Okay, CO2 assimilation, that is represented by AN. AN means assimilatory rate. Got it? So these are 4 or 5 basic parameters that you can get from IRGA. Apart from that, you can go for ACI curve. ACI curve means, A means already I told you, assimilation rate. Okay, so in x-axis this will be assimilation rate, in y-axis this will be your internal CO2 concentration. Got it? So usually what is the atmospheric CO2 concentration in ppm?
300 to 410.
Around 400. Got it? So atmospheric CO2 concentration is 400 ppm. So if we increase the CO2 concentration or you decrease the CO2 concentration, okay, what will be the effect of plant if you want to check? For example, if a plant is growing continuously under 400 and again you are shifting it to from 400 to higher concentration, let's consider 800 or even 1200 or 1600 or 1800 ppm. So that way how the plants are behaving. Okay, and again in the higher concentration to the lower concentration, how the plants are behaving, you can measure through this method. Got it? So if you have any stress kind of plants, and if it is related to your CO2 uptake, how the CO2 fixation is there, how the stomatal conductance is operating, how the net CO2 assimilation is there, how your Rubisco activity, how is it happening, that can be measured directly from this. Actually, ACI curve, through ACI curve, you have to first, I'm not going in details with the ACI curve, because if I start ACI curve, for one sample, it will take at least 45 minutes. Okay. So first of all, for taking IRGA curves, it has to be, the leaves has to be always connected with your plant. Once it is detached, the water content will drop. And once the water content drops, everything else will be hampered. Transpiration rate, stomatal conductance, net CO2 fixation, everything. Because ultimately through stomata, the water vapor evaporates. So if the water vapor is less, or if you are plucking it, the plant leaf is having a molecular mechanism that it will close the stomata to just prevent the water loss. So once the water loss is less, you will get faulty readings, because stomatal closure will be there. Once the stomatal closure will be there, CO2 fixation rate will also drop, and also net CO2 assimilation will also drop. Got it? And similarly, because stomatal conductance is directly correlated with your transpiration rate, that will also drop. Got it? So this thing, one thing you need to be always clear is that the leaves should be always intact with the plants, that is in vivo measurements. Right now, I don't think IRGA has for algal samples, but for leaf samples, you can do it. Got it? So, any doubts up to this? So how does it measure? First of all, we have to give a reference CO2 concentration. You got it? So it basically measures your CO2 concentration. So if we give a reference CO2, for example, the atmospheric CO2 concentration is 400 ppm. So we'll give you a reference CO2. Okay. From the reference CO2, if a plant absorbs some amount of CO2, if a plant absorbs more CO2 concentration, if a plant absorbs less CO2, then the reference CO2, comparing to the reference CO2, we can know how much the decrease is there. And depending on that decrease, we can say the plant is performing well or it's under stress or any kind of thing. Got it? So first, you need to, so for general photosynthesis, what is needed? Normally, what is needed? Normal photosynthesis. CO2, then O2 is actually not needed. O2 is required for respiration. For just photosynthesis, what is needed? First of all, water. Okay. Pigments, already it's done. Pigment protein complexes are there. Then what is needed? Solar light or light, actinic light. Okay. In the form of, in nature, we get it from solar energy. So this exactly does the same thing. So already you have the plants. Okay, plants, already the water, everything is there, the chlorophyll. Only thing, because we are performing through manual method, we have to provide a light source. Got it? There is no need of dark adaptation because we are not measuring fluorescence. Okay, so fluorescence, because we are not doing, there is no need of dark adaptation. But to stabilize the readings, it will take like 4 to 5 minutes. Okay, for one reading to stabilize. Normally within 3 minutes the readings get stabilized. So this part is your chamber, IRGA chamber. Okay, so here you see there are two pipes. Okay, one will be for reference and one will be for sample. Okay, reference and sample CO2 difference will be reflected in this machine. Reference will be a constant CO2. Usually if a plant is constantly grown in 1400 or 800 or lower CO2 concentrations, even 200 or 100, that will be the reference CO2 concentration of that plant. Got it? If it is grown continuously from the starting point. Okay. So if I got this from outside, so outside the surroundings, what is the CO2 concentration? 400 ppm. So we have to keep here 400 ppm. Until and unless you want to check how the plant is performing in very high CO2 or very low CO2, the ambient CO2 or the plant condition in which the CO2 is there, whatever the CO2 concentration is there in that ambient condition or in which the plant is grown, that is taken as your what? Reference CO2. You got it? And sample CO2 will be based on the plant's photosynthetic performance. So sample CO2 will never be higher than your reference CO2. Remember this. Sample CO2 can never be higher. You are supplying 400 ppm. So will it go 500? It can never go 500. Only the CO2 has to be utilized. The utilization might be less or the utilization might be more. If it is less also, it is never, at least 400. 400 is also very uncommon. 400 means the plant is not at all fixing anything. So it is under huge stress or might be the plant is dead. Okay. So just remember one thing, reference CO2 concentration is always higher than your sample CO2 concentration. Okay, that is internal CO2 concentrations. Another thing what you need to do is, these two are for, one is for reference CO2, one is for sample CO2. Now how we supply CO2? We supply CO2 with cylinders. Okay, these CO2 cylinders, we use it to supply the sample CO2, sorry, reference CO2 concentrations. Why this 12 gram CO2 cylinders we use? Usually it will last for 9 to 10 hours. Then you have to discard. Whether you do the experiment or not, the CO2 will be released. Got it? So once it is punctured, it has to be used in one go. So if you want to repeat the experiment tomorrow, you have to use a fresh CO2 can. In some people who can't afford this CO2 cylinder, these are very costly, around 500 to 700 per capsule. So if you are not going for this, there is an other way, but in that condition you cannot go for high CO2 concentrations. It will get the ambient CO2 concentrations. So for ambient CO2, if you are using ambient CO2, there is a hole here, this one. You can come here and see, or else this one. Okay, so if you are not using this CO2 cylinder, you can directly put a pipe here. okay, and that pipe you can put in a thermocol box. And why in a thermocol box? So that there is no pressure fluctuations, okay. Air pressure fluctuations, if it will be there, then CO2 will be more or less. So we have to just maintain the whatever the pressure is there. So that's why you just connect it to a thermocol box and just cover it slightly, just open the box and it will maintain your ambient CO2. But if you want to go for very high CO2 concentrations, more than 400, 600, 800, 1200, then 1600 or 1800, then you can't go for that. For that you will need this one. Got it? That is the reason we use this one, because we use both ACI curve as well as APAR curve. APAR is CO2 assimilation and PAR is photosynthetically active radiation. So with respect to PAR, your CO2 assimilation will be plotted. APAR. A is in y-axis, PAR is in x-axis. Similarly, ACI is A is in y-axis, CI is internal CO2, that is in x-axis. Got it? So first of all, we need to puncture. Got it? So before that, we need to fix this chamber with the analyzer. So this is basically the analyzer. The name is Lycor. Okay, Lycor, and the system is 6400XT. Okay, and this is a portable photosynthetic system. There is another system, more developed one, that is Lycor 6800, and that is more portable than this one. Got it? So this part is your analyzer and this one is your IRGA chamber. So what does the chamber consist of? The chamber here, if you'll see the top here, LED source is there. Okay, apart from LED source, infrared radiations are also applied. Got it? Why the infrared radiations are applied? I already told you, it has to detect the heteroatomic molecules, that is the CO2. Okay, only CO2 it will detect. So once the CO2 is there, whatever CO2 levels are inside this chamber, it will compare with the reference CO2, and then if it is dropping, then how much drop is there, that it will calculate and show it here. Got it? So LED source is over here, and there is a small wire like thing that will maintain the temperature of the leaf. Okay, because leaf temperature is also directly proportional to your stomatal opening and closing. Got it? So you want to make sure that the leaf temperature does not fluctuate too much. Okay, so these are all the gaskets. You just have to place your leaf here and close the chamber. So for closing, usually to avoid damage to these gaskets, we loosen this when we store it. Loosen this means there will be some gap, okay, between the gaskets. So while using it, make sure you just tighten the gaskets first. So to tighten it, just turn it around. Yeah, it is more or less tight. Okay, so once it is tight, you can just go to any light source and see whether there is light leakage or not. It should not be too much tight. If it is too much tight, then you will damage the leaves. Okay, and it should not be loose. If it will be loose, then CO2 will escape. Your reference CO2, whatever you are maintaining, it will escape. Got it? And there is also a light detector. This is the ambient light detector. Whatever the light means PAR intensity is there in the surrounding, this will detect. Okay, and apart from this, these are all to cool down everything. LEDs are there, these are all the fans to cool it down. Just these are your basic things. Okay, this is the main chamber. This is for your one is for your reference CO2, one is for your sample CO2. Got it? So once this chamber is done, this part is your analyzer part. Okay, so this is the main component. Here all the analysis part will be done and everything will be recorded here only. Got it? So first you have to connect this with your chamber with your analyzer. So for this there is cable. Okay, yeah, you can use it. So here you see there is a black colored mark. Okay, so this black colored has to be matched up with this black. Otherwise, if you will mix up with reference with sample and sample with reference, all readings will come faulty. Okay, so first thing you have to do is to fix it. And another thing is that these gaskets, once these gaskets are damaged, you have to replace it, because if you will not replace it, the CO2 will leak, and once the leakage will there, again the readings will come faulty. Okay, so by the time he fixes, I will tell you what exactly just fix. Okay, so this is your main analyzer part. So here the CO2 cylinder goes. Okay, See here, there is a rubber gasket. If this gasket is even damaged, you can remove it with the help of, there is a prong here, just you remove and you can insert a, typically after every 4 to 5 readings, you have to change the gasket. It's not damaged, so you can just see whether the gasket is damaged or not. Just physically you can see whether it is not damaged now. Okay, I replaced it recently, so it is not damaged, but typically 5 to 6 readings or even 2 to 3 days you have to change the gaskets. Okay, so you just put it inside. So I will just loosely tighten first. Once these are connected, then I will tighten it, because there is also a chance to the CO2 leakage might be there from this way. Okay, so I will just loosely tighten it. Now the puncture is not there. Once it will become tight, and you have to give a final tight one. That time the puncture you will get a very small shush kind of thing, like air escaping. You will get some kind of thing. Okay. So apart from this, there are two. These two are, one is your dryite and another one is your soda lime. Okay. So if you are using CO2 can cylinder, it's basically first this CO2 is under huge pressure. Got it? So a gas under huge pressure becomes in liquid phase. Okay, so gas under huge pressure, it comes under liquid phase, basic physics. So now if it will release at the same time, some amount of liquid water vapor will be there in it. Okay, so yes, moisture will be there. So in order to remove those moisture, you have to scrub it. Okay, so for scrubbing you have to use what? Not silica. We don't use here silica. We use here soda lime. Got it? Soda lime. This one. We use this one, and for daylight to dry out whatever the another thing. This color of dryite you see, which color? Blue color. Once once it becomes dry, once it becomes moisturized, it will turn to pink color. Okay. So once it completely turns to pink color, you can't reuse it again. You have to again heat it. Okay, heating may be done for 3 4 5 days at 100 degrees in oven. Okay, after that all the water vapor will evaporate. Many labs do that if they can't afford to buy every time. Okay, so you can reuse 2 to 3 times, not more than that, if you really want to. Okay, so these are the two things. First, let him connect. I will tell you. This is your soda lime. Soda lime is here, you see. CO2 scrub is there, na? Here you see what is desiccant. Desiccant will basically, it will your take surroundings, it will have water vapors in it. Even this one also. So it will dry the air. Only CO2 will be taken out. Got it? And this is also to remove whatever the moisture is there. Just to clean the air to get pure CO2. Okay? The function of these two is to clean the air and to get pure CO2. Connected? Okay. So see this connection, keep it well. Okay, you see, one is your just the basic connection. There is no big deal in it. First, these two connectors will not interchange when over there, because only one connector is that side and two connectors is here, and one is female, one is male. So you will not confuse which connector is what. Got it? So these two are for just to connect these, all these things. Light source is there, fan is there, to everything, to connect everything. These two are there, right? So now you see these two pipes. These two pipes, one I told you, it has to be same for everywhere. There are also two pipes. Here also two pipes. One is for reference CO2, one is for sample CO2. The protocols you have, na, that is shared. Protocols is not shared. Okay, we'll share it. So once this thing is done, you have connected everything. First you have to do is to battery nahi. So this is actually a portable system, but it is actually very heavy. So anybody wants to just try how heavy is it, they can try. So this is just your this purpose, okay? Maybe more, 15 maybe, and if you will include these and batteries, it will be more than 35-40 kgs. So put it inside, no? Put it inside. So batteries, these are rechargeable batteries, okay? So depending on what work you are doing, it will last for max to max 3 hours, okay? So if you are doing general parameters, it may last for 4-5 hours, but if you are taking ACI or APAR curves like 6 to 7 readings, if you are taking in a day, they will last for 2 hours max to max, okay? So some people, they use direct AC source. So you can connect in these two batteries, you can connect a AC source, but the connector has to be this type. Got it? So once you switch on this, pull over these batteries, then switch on the instrument. After you switch on, the initializing program will start. Okay. Okay. Once you start it, the 2x3 LED light source, you have to connect it. Okay. Not the factory default. So 2x3 LED source is because of 3 centimeter in 2 centimeter. This one. Okay. The size of this one is 3 centimeter, 2 centimeter LED source. So you have to first connect it. So you just select. Okay. Now it's asking, is the chamber or IRGA connected? This is the chamber of the IRGA. So first of all, you have to make sure it is connected or not. Just make sure everything comes. Okay, there is a black ring around it. So this will go with this one. Got it? There is no black color anything, just a ring around it. So that means, okay. So you just press yes, Y for yes, and for no, if it is connected. If it is not connected, then you have to press no. So you see the sound, some click click sound came, right? This is the mixer. So it will just test if the mixer is working or not. Mixture, what happens if you use this CO2 cylinder, it will mix this CO2 with some of the atmospheric CO2. It will not utilize everything. So that, with the help of that, this mixture operates. Got it? So if it starts to utilize CO2 everything here, maybe it will not last more than 1 hour. Okay, that is the reason this CO2 mixture is done. Now this thing has started. You will hear a fan sound.
So then the concentration may differ.
Why? No, we have to give a reference CO2, na? It will adjust accordingly. If you are giving 400 ppm, see 400 ppm we don't have issues. If you want to go for higher ppm, 1800. So the atmospheric CO2 is 400, right? It cannot go to 1800. So it will take some of the atmospheric CO2 gas and again it will mix the CO2 from here. This is complete CO2. Okay. So here, and it is under pressurized form. So you just pressure some amount of CO2 it will take from here and some 400 ppm from outside. It will mix and make it up to 1800. Got it? This is the reason this mixture works. So when you will start the IRGA, if the mixture is working properly, this click click sound initially it will come. If it is not working, then the mixture will not work. Okay. So once it has started, I will start to puncture it. So it is done. Okay. So the puncture is done. Okay. Now what you have to do? First, if I take any light intensity, for example, if I take 200, so will I be able to get a maximum CO2 assimilation rate of this plant? No. Why? Why? No, it's not our reference CO2. Let's keep reference CO2 as 400. Okay. So if I start to give a 200 microinstein light intensity and I record the readings, there will be some readings in 200. So will it be the maximum CO2 assimilation rate of the plant? No. Why? Why? I told you already, maximum CO2 assimilation rate. Maximum CO2, you know the law of limiting factor? What does it mean? Means? Yes. Not even, not reagent, anything can be lower. Yes. Any component, whichever is lower, that will directly affect the product. Okay. So now, if the plant is already grown in 1500 microingstine, 1200 microingstine, and you are giving rest all the parameters same, CO2 you are keeping same, relative humidity you are keeping same, light apart from light, but light you are giving 200. So will it show the highest CO2 concentration? It will not show, because it is already grown in 1500. So for that you have to go for a par curve. A par curve, once the curve will come, na, there will be a point in which there will be a drop. I will show you how the A par curve is plotted. So every plant will have a saturating light or par intensity. If you go beyond the saturating par intensity, there will be photo inhibition. If you go lower to the saturating intensity, it might be limiting. Limiting means the plant might not be performing to its full potential. Got it? So that's why you have to first give a saturating par to the leaves. For that you have to determine which is the par. Got it? Which par intensity should be given. So for that you have to first perform a par curve. Okay. So for a par curve you just have to press here 5. Okay. Just press here 5 directly. Sorry, you go to new measurements first. First will be new measurements. Okay. So once the new measurements will come, this thing will start. Okay. Then you just press 5. Okay. From here, another thing, before running, you just see these readings. See these readings. So you see CO2 reference and sample. Did I switch on the mixture? Nothing. I didn't give any values of CO2. So everything should be, that means leaves are there inside it, no leaves are there. So it should come around 0. Got it? So photosynthetic rate before starting, this should be around 0. You have to wait for 5 to 10 minutes until this warms up. So it will take like 10 minutes to warm up and it will come around 0. Around 0, it may not be exactly 0. It will be around this kind of, it will increase, then it will decrease. Got it? So for that, until that time, you have to stay. If it does not come to 0, then you have to again recalibrate it. Okay. So if it is required, I will tell recalibration. If it is coming to 0, then no need of recalibration. So you just wait for 10 minutes. So make sure that this is closed. So see the difference if I open it. Just see CO2 reference and CO2 sample. Okay. So if I just open it, see the sample CO2 concentration. Got it? Then this CO2 concentration is increasing. Okay. So you have to use it first. So how do you know at what time it is getting stabilized? The photosynthetic rate should be 0, around 0, 0.01 something, or minus 0.0, that is also fine. But it should not be 1 or 2, 3, it should not be like that. So first there row, the first row is okay, second one is CO2 sample. So another thing is that before taking any readings, we have to match the CO2 reference with CO2 sample. But before matching, we have to wait until the ergas become warmed up. Okay, it will take like 10 or 20 minutes to warm up. After this, you can matching. Okay. After this matching, this is your water reference and this is your water sample, and this is your change, net change. What is the change in reference and sample? Here it is reflected. Okay, it should not be negative because we have not kept any leaf and this is not calibrated. That's why it's showing negative. Okay, once it comes to 0, this one will come around 0 also, and this is your also change in the water, this too. Okay, these most of these two will be same, but major thing we have to calibrate this two. Reference CO2 and sample CO2 has to be same. Then this is the flow, relative humidity should not exceed 75%. And then transpiration rate, this is internal CO2, this is conductance, this is net CO2 assimilation, okay, photosynthetic rate. So I think that's fine. Okay, so you see now the readings are more or less stabilized. 0.41, 2.42, it's not that much. It's not going 3.5, 3.7. It will come like that. If you want to go for graph, you just press this end. Just you press this. Here you will see. Here the saturating point is already reached. This point. It was increasing. Then the saturating point has reached. So now you can know that readings are more or less stabilized. Got it? So you just press escape. Once you press escape, it will come back to your main menu. So it is 2.45. So it is not calibrated. So first we have to do is again calibrate it. To press again. Okay. Then menu. Okay. Then you go to IRGA. Then you go to. Then press enter. And closed, okay, and total M desiccant, it's like 4 or 5 times it has been used, so it is fine. Okay, now we have to press Y to continue. So then again it's asking the chamber must be close to 0, CO2. Press any key. Everything is mentioned here, no need in too much, because it will be whatever the readings will need, it will in here. So you have to press any key, just press any key. So it will take some time to zero the IRGA. Okay. So these two readings should be around same. It may not be 0, but the reading should be same. So what do we have to do? We have to make this 2 sample as 0. Okay. So initially when you will wait, this readings will stabilize. It will be minus 5.5, 5.6, 5.7. It will be 9.1, 9.2, 9.3, 9.4, this kind of thing. So after that, these values are a bit different, right? Then you have to zero this. Zero this means these two values will come to closer to each other, very close to each other. Okay. So you can just go to CO2 reference sample 0. CO2 RS means R for reference, S for sample. So these two things you want to make it to 0. Got it? So you can just go here. So it's not changing much, you see, 9.2 to 5 minus 5.6. You just press F1, just the beneath this button. So setting 0 low, setting 0 high, then it will make two values almost similar. Okay, now you see. So the reference and sample CO2 is around 0. Okay, 0 0.0, 0 0.0, around minus 0 0.0, 0 0.1, even sometimes it happens. Got it? So once this is calibrated, you have to quit. So now if you want to just press escape, again you go to new measurements, then you see these values will be almost similar. Okay, so now reference CO2 and sample CO2 will be, and here also the photosynthetic rate will be, should not exceed more than 2 or 1. Got it? So again you have to wait for like 5 or 6 minutes. So if you want to really check what is the plot, just press this one and it will show the plots thing. So until and unless this line comes in a straight line, so that is the saturating point. If it is dropping or if it is increasing, then you will know that this is not the proper time to record the readings. You have to wait until these readings stabilize. Okay, not saturate, stabilize means it will come in a almost more or less straight line. So almost it has stabilized you, so it's not changing too much. This is also a bit straight line also. Okay, so you see the range is from -1 to 0, so it's very less range. So it is straight line only. If you just compare from 0 to 100 or 0 to 50, this will be completely straight line. Got it? So now once the IRGAs have warmed up, just go to escape. Okay, so also you see the photosynthetic rate has not changed. Got it? Now we have to go for APAR measurements. APAR will determine your maximum photosynthetic active radiation. Okay, so you don't want to give a light that is photoinhibitory to a plant, and if you don't want to give a light that will record or down record a photosynthetic rate of a leaf. So first you have to measure the exact PAR intensity. So for that you just press 5. Okay, then you go to auto program. There is an auto program based on it. So you go to a light curve 2 here.
Sir, 5 or F5?
5. After F5 there will be direct how to, I'll show you again. Just go to 5. This is 1. Okay, 2 comes this one, 3, this is 3, this is 4. Okay, this is 5. Got it? So before going to 5, let me tell you one thing, since I'm browsing everything. So you leaf fan will be fast. This fan will be fast. So flow is 400 micromolar. Mixture is off. Now mixture, since we use this one, we have to give how much? 400 ppm. So we have to add the mixture. So reference U2 is already, it is set as 400. Got it? So you just keep means it will keep this thing. Got it? So reference U2 is 400. So temperature is off, lamp is off. In APAR, the lamp will be automatically it will start from highest intensity and it will go back to lowest intensity. Got it? So you don't need the lamp because you have not decided which is the PAR, exact PAR. Got it? So it will automatically increment from 1800 or maybe 2000. From 2000 it will start to 1800, 1400, 1200. That way it will check at what intensity there is photoinhibition and what intensity is the perfect to take the reading. Got it? So you just press, now you press 2. This is the second one. Okay, so if you go to 3 usually marks the area of the leaf. You have to give either you can take a graph paper to plot the exact area, or else you can averagely calculate it. It's not, maybe it's not 6, so I may write it as let's say 5. Okay, 5 centimeter square, then press enter. This is the average one I'm taking, this leaves. Okay, so apart from this is 3, this is 4, this is 5. Okay, so this point you need auto program. So once you go to auto program, click it, then light curve. So after you press the light curve, we have to give a name of the file. Okay, so first I am giving a name of the file, APAR space DEMO 1 space, okay, demo, just write GIAN. Okay, so this is your file name. Okay, APAR demo GIAN. Just you press enter. Okay, a remark you don't need to write. Press enter. Now you see the summary is 12 points. Okay, 12 points means the light will be decreasing from the highest intensity to the lowest intensity in 12 decremental values. Got it? So the first value will be your 1600. Okay, 1600 PAR will be your first PAR intensity, and the next PAR intensity will be like check. Okay, so first PAR intensity will be your 1600, then comes your 1400. This you can adjust manually also. You can change here if you want, but it has to be which one? First PAR reading also you can change, but usually it will not go beyond 2000. 2000 is more than sufficient PAR reading. The sunlight is around 1800. No, you have to determine a PAR. Let's say example, if I just write 1800 here also, it will start from 1800, then 1600, then 1400, then as long as you get a saturating point, there is no problem. If you don't get a saturating point, you may increase the PAR, but maximum it can handle 2000 because the sunlight's PAR intensity is 2000. Got it? around 1700 to 1800 natural sunlight. So it starts from 1600 power, then it goes to 1400, 1200, 1000, 800, 600, 400, 200, 10, 50, 25 and 0. Okay, these are all the power readings. So you just go to escape because I didn't change anything. So each point will last for around 180 seconds. 180 seconds means consider like more than 2 minutes. 2.5 minutes, how much is 183 minutes? 3 minutes. So max to max it will take 3 minutes. Within that it will take a point. So each 12 point means 1 point is 3 minutes. Understood? So you just do 12 into 3. How much? This is just for taking one readings. So you have to wait for 36 minutes to check the exact power intensity that a plant is performing well, or a photoinhibitory radiation, you can know. Okay. So we'll just start it. Once you start it, before starting, you have to keep the leaf. Suhail, bring that one. Okay. There is tissue now. And make sure you wipe the leaves. Otherwise, yes, because of the dust, it should be fine.
They don't like water if they have
Water content is also there, na? Means it will directly affect your transpiration rate and everything. So just slightly just wipe the leaf. So while placing the leaf, make sure that the leaves does not fold, okay? Because if the leaf folds, your values will come different. Single leaf, it should not be overlapped. Okay, so you can start it now. Just wait for 3-4 minutes, or else you can start the readings. Okay, now the reading has started because this star mark you see it's blinking. Okay, this star mark you see it's blinking. First 2 seconds it will blink. By the time you saw it stopped. So you see now the photosynthetic rate will be in positive side. Okay, so it's coming to like, it will record. It's not stable yet. So if you want to really see at what point it's coming, what, just press this one. So it is still not stable. You have to wait. So just to view the graph, you press this one. Okay. 5 star means sometimes it's blinking, na? That means the program is running. Okay, so this now it has almost stabilized. Okay, now you have to go to scroll these things and go to your this one. Okay, A par, this one you have to go. Now it is around 1600 around. So the photosynthetic rate is very less. Why? Because we shifted from a lower CO2, outside CO2 is 400 to here, that is very high. Okay, that time I opened and show you, right? It was around 1600. So that might be the reason the stomata is closed. Okay, but usually natural conditions it should be around 4-5 something it should come. Okay, so now if you want to see what point it will come, so you just wait until 3 minutes it will take, and it will go to 0. Definitely it will go to 0, but you have to wait until whatever point it drops below 0, that will be your compensation point. Means beyond that, if you give light, the plant will photosynthesize. Below that, the plant is not able to photosynthesize. Got it? Because the CO2 inside this, we got it from outside to inside. That was a shock for the plant. So usually it is ideal for the plants to take where the plants are grown. Just for demo purpose and this video filming, I got this inside room. Got it? Another thing, at each point, whenever the plant takes a particular condition, this point is recorded, right? Okay, this point is recorded. Now we don't know 1600 might be photoinhibitory or not. If this point is higher than this something 1 or 2 just coming initially. If it is higher, then it is not photoinhibitory. If it is lower, then it if it is higher, then the next point is photoinhibitory. Let it run. I will tell you exactly the graph you can get. So this was your 1600 power, 1599 written, right? So last power we gave is 1600. So now the next power is running 1400. It's running now. Got it? So until 3 minutes, it will wait to see the readings are getting stable or not. After 3 minutes, it will take the readings automatically. And every readings is followed by a what? Sampling of the water and CO2 reference. Equalizing the CO2 reference and sample, every reading has to be done. So this is the basic idea. The actual photosynthetic rate will go beyond 10, 11, 12. Okay, if the plant is in native place. If you shift the plant somewhere else, then for maybe for temporary time, it will close its stomata because you are shifting from low CO2 to a very high CO2 here. Got it? So that might also stress out the plant. So the second reading is taken. You can see here the CO2 reference and sample is almost same, 400 ppm. Okay, so these two readings are already taken, these two, and it is very low. So if you can see, this point is slightly lower than this one, right? Slightly lower. So 1600 is your photoinhibitory, at least till here. We don't know about this point, what is going to happen, but this one is 1600 is definitely photoinhibitory. So we have to wait whether this point is higher or not. If it is higher, then this point is also photoinhibitory. So you have to take this point. So again, if previous power intensity is higher than this, then this point is also photoinhibitory. So you have to see what point is your photoinhibitory point. So you have to take that point which is not photoinhibitory and which gives a highest point. Okay. Okay, you can sit. By the time it will take it will plot all the graphs. Okay, a lot of time. APA they have to do first. We got in inside plants. We got the plants inside. The CO2 concentration is very high. Inside, people are here, na? That's why it's even very less for the take rate. Who is the fans? Of the fan.
So what is the cost of this instrument?
Updated instrument is also there. Updated at 6 8 6.
టూ టైమ్స్ ఇచ్చాడు అక్కడ ప్రొసీజర్ అందుకే అడిగించండి. AN is equals to 0 after putting the leaf in the chamber. Again wait until AN is equal to 0 అని వచ్చి టార్గెట్ ఏంటిదా పీటీ టీ టార్గెట్ అని ఇస్తున్నాడు కదా అదేంటి ఇంకా వెళ్ళలేదా సార్ వెళ్ళిన రోజు
ప్లస్ 5 ఫ్రమ్ ద కీప్యాడ్ ఎందుకు న్యూ డైలాగ్ బాక్స్ ఓపెన్ అవడానికి.
ఈ పర్టికులర్ ది 2 సార్లు వచ్చింది అని అడుగుతున్నాడు ఎవడు అంటే
Putting beyond one, so you will not get a graph. It is kind of 10 or something, then you find it. Because if it is coming to 1 to 0, it will not come as a total. So that might be a different. Yeah, we can do, but inside this condition, I don't know. Data is very specific. Also, that, but that's why these are also different. Pressing no, it will ask will be continuing on, but before connecting the IRGA, put the Li64 into sleep mode. Sleep is found in until menu. Okay, so no need of doing, just press enter to it. Just go. So again, it will now come back to the main menu. Connected, na? Okay, so first you have to connect here L term. L, you have to press L, then you have to press your T here. So first press your L. So once you press L here, it will be shown as L term. In the home menu itself, it will be shown L term. Now just open this one, Li6400. This one T is written, right? Open this. Then you have to select which connection is needed, because this one is RS232. It's not Ethernet cable. Okay, you have to select RS232. Okay, and only one communication port is available here. You just go to connect. Okay, now this is your IRGA. Whatever the IRGA is here, it's displayed here. Got it? It is just mirroring whatever things are here over there. So now you go to that. Yeah, you have to go to files. Okay, so right side you see there is a plus sign here. Got it? Just click on it. Then go to users. Again press here. Okay, once you press on users, all the files will come. Okay, whatever name you have given. So I'll just browse and see whether today. Okay, here you have to only take the .xls. Okay, so you just take it, drag it and drop it in whatever position you want. I dropped in downloads. Okay, and apart from that, I don't know whether the other thing recorded or not. Okay, I am taking another one to show what exactly how it should look. So here 60% you are seeing, right? 80%, that is the transfer file. So once it is done, you can go for next. So just for showing purpose, I am just taking one of my files. And this one also, I am taking one of my files. So only .xls is the Excel file. It can only open in Excel file. Other files it won't open. So you just copy only the .xls files. Okay. It's not increasing to 4. Okay, it should increase. It's not increasing. Okay, once it is done, just close the file, then you go to disconnect. Okay, just go to disconnect. Once it is disconnected, you can close the software. Yeah, and after that you just press again L, so that L term will disappear now. Then you can switch it off. So now coming to the plotting thing, I'm showing how to plot. Okay, it's corrupted. Maybe okay, the file is somehow corrupted because I stopped. Anyway, I'll be showing you how the file looks like, exactly how the Excel file looks like. I have one of the Excel files. GS is your stomatal conductance. Just divide 13.575 divided by 0.29. Okay, so whatever value it comes generally goes with your supplementary data. Okay, so the reviewer might ask why you have taken this per intensity, why not more or why not less. So that way you can show that at this point the highest for that, if CI is less, then the plant is good condition or not. You have to check along with it the stomatal conductance as well as transpiration rate. Obviously two things go hand in hand, one is stomatal conductance and another one is transpiration rate. If stomatal conductance decreases, transpiration rate will decrease. If stomatal conductance increases, transpiration rate will also increase. Fine. Okay, E is your this one transpiration rate. Okay, E is in paper format, E is represented as transpiration rate, G is stomatal conductance, Ci is internal CO2, N is your CO2 assimilation rate, and WUE, that is A divided by GS, is your water use efficiency. Okay, X-axis depends on whatever conditions you take, wild type or treatment plants you can take. Okay. Okay, I think this is done for today. Anyone has any doubts? Okay, fine. Thank you.